So welcome. This is our annual INSAR pre-conference meeting this year in Montreal. So welcome, everybody. I am going to try to see if my stuff works. Perfect. Oh, this is an awkward screen. OK. So I thought I would start today with, I'm Dr. Anagnostou, by the way, Evdokia Anagnostou. I'm a child neurologist at the University of Toronto, and I'm the chair for this year's meeting. And I'm thrilled to be here today for the local pre-conference meeting. So I thought I would introduce a little bit of INSAR first, so that you guys know what this event is about, and then I'm going to introduce you to the speakers, and then we'll talk a little bit about some Canadian updates, and then we'll get into the meat of the program. So INSAR is the International Society for Autism Research. It's our scientific and professional organization. It does run an annual meeting every year in the spring, and the purpose of that scientific meeting is to exchange scientific information between the different scientists, researchers, and professionals from all over the world. So this year's meeting is the largest ever in the history of INSAR. We have 2,400 and something researchers and clinicians from all over the world in the city, and the meeting is going to start tonight, and it's going to go all the way to Saturday. But there is this tradition of the INSAR pre-conference, which is the local community meeting that happens the first day of the main meeting. And its purpose is to showcase some of the international speakers from the main meeting, as well as some of your local scientists and well-known researchers and clinicians. It's supposed to be accessible to the community, so it's open to the public, unlike the main meeting, and it is an opportunity for the city that the meeting is run in to engage with the autism researchers. Now, it doesn't come often to Canada, and so we didn't want to limit this to Montrealers or Quebecers. So we have a sponsor, the Ontario Brain Institute, you're going to hear later, that's funding a live feed. So right now, we have people all over Canada registered who will be watching the morning session. So very quickly, let me introduce you to the keynote speakers for the morning. We have Dr. Steve Scher, who is a professor of genomic sciences at the University of Toronto. He is credited with the discovery of copy number variations and their impact on health and disorder, and the contribution of those to autism and related conditions. And he is a very well-known, very well-published, more than 500 publications on his record, and very well-awarded scientist. He just won Canada's Killam Prize to medical science, a very prestigious award. So we're thrilled to have him. He will talk to you about updates in genomics, both within the Canadian context, but also internationally. And then we have Dr. Jason Lurch from Oxford University. He's the director of the preclinical imaging there. He's also an associate professor of biophysics at University of Toronto, so he's commuting back and forth from Oxford to Toronto these days. And he is doing a lot of exciting things, but the things that he's going to focus today is help us think through how we can use animal models to understand how brain develops differently in developmental conditions, and in particular ASD. Then we have Dr. Elizabeth Berry-Kravis, who is coming to us from Rush University in Chicago. And she's a professor of pediatrics and neurological sciences and biochemistry. And she has spent the last 30 years studying rare neurogenetic disorders that are associated with neurodevelopmental conditions. And for the last few years, quite a few years, she's been working on translating our understanding of this molecular biology to novel treatments related to autism and other neurodevelopmental conditions. So we're thrilled to have her. And last but not least, we have Dr. Diana Robbins, who is a professor at Drexel University in Pennsylvania. She is working on understanding the best ways to screen for ASD and early detect to facilitate early intervention, which I understand is a particularly hot topic these days in Quebec. So we're looking forward to her. She has developed the MChat. She's the developer of the MChat, and she will present a lot of work to make us think about universal screening versus surveillance for kids with ASD. In the afternoons, their speakers will introduce themselves to you at your workshops, but we have a group of very well-known Quebec scientists for you. You have already picked your workshops. So Dr. Aparna Nadig is going to lead the bilingualism workshop. Dr. Sulia and Obino would lead the workshop on inclusive education. Dr. Baudois Forgeau Dark will lead a workshop on mental health. Dr. Eve-Marie Quinten is going to lead the workshop on the effects of music on social, cognitive, and brain functioning in kids with autism. And last but not least, Dr. Flanagan and Dr. Meng-Xuan Lai will lead a workshop on inclusive identities, ability versus disability. So this is your schedule for the day. But now I want to introduce to you some very special colleagues who are with us today. So we have 3 autistic artists who are here to participate in the meeting, but also showcase their work and their talent. And I'm going to start from River, Christy White, who is a young First Nations dancer. River had this very traumatic experience where he lost an autistic friend to suicide, second to in context of bullying. And so he's using his art and his music and his tradition to inform and educate both First Nations but other communities about stigma and autism and ability and inclusion participations. So he will perform up here his dance at 3:30 after our workshops. I'm thrilled to have him, and I hope you all come back on time to enjoy it. It's really a treat. Then we have Mr. David Beresford, who's sitting on the back, and he's waving at you. He is a brilliant caricaturist and illustrator, and he is also an autistic person. And he's showcasing his work both in the world of arts and in the world of autism. And he's going to showcase his work and sell his work later, so please stop by. And last but not least, for our artists, I have Miss Ruby Stevens, who I am thrilled to bring here. She's a young lady that I know very well, very gifted artist with an interest in the area of anime. So she has brought her work for you. She's also a competitive swimmer, and she's on her way to the Canada Paralympic team for competitive swimming. And then there's you. So I want to let you know who you are in case you don't all know each other. So there is actually 288 of you registered in person here. And you are professionals, you are families, you're autistic self-advocates, you are graduate students, and we're thrilled to have you all. But we also have, as of last count, more than 120 people logging in from all over Canada, even from Yukon, attending, mostly families and some professionals for the morning sessions. All right, Canada quick updates. So we finally have our first Canadian epidemiological study funded by the Public Health Agency of Canada. So now we have Canadian numbers. In case you are curious, we're sitting at 1 in 66 kids having a diagnosis of ASD. Our numbers are very similar to other Western countries, especially the US. There is a little bit of variation from province to province if you get to look at that report, but I want you to think that there is actually variation on our healthcare systems from province to province, and so access to diagnosis and service delivery is provincially mandated, and therefore you'll see those kinds of small variations. Quick update on research. So I thought I would just highlight the commitment that Canadian agencies have made to autism research over the years. So I will start with the top, the Tri-Council funding. So for those of you who are in research know what I'm talking about, but for those of you who are joining us for the first time, these are the agencies that provide federal funding, most of the federal funding for health and social sciences. related to what we do. So over the years, the total amount has been more of $100 million commitment to autism and related conditions from Tri-Council. The number from Genome Canada is about 96 million right now, so another very large commitment that has come through for understanding the genomics of autism and related conditions. Now, the next category, I had no way of getting their full amount, so I'm gonna show you only competitive funding for the last 5 years, right? So this is not the enormous contributions these organizations have done, and I'll highlight the exceptions in a second. So the Ontario Brain Institute over the last 6 years has committed 31 million to autism and related conditions. Azrieli Foundation is in the room, and they have committed north of $50–60 million. This is just the number for competitive funding over the last 5 years through the Brain Canada match. The Simons Foundation funds research all over the world, but about 3 million of that over the last 5 years has flown into Canada. And again, Autism Speaks has funded research all over the world, but about 2 million of that has flown into Canada over the last 5 years. So even when we restrict what we examine to just competitive funds through traditional mechanisms, there has been a significant investment in autism research in Canada. There are many large initiatives. I have only examples here, and mostly examples of people who may be represented in the room. So missing, you're gonna hear quite a bit about from Dr. Sher. It's a very large international autism genomics initiative led by Canadian groups. Kids Brain Health, funded by the National Centers of Excellence, has looked across neurodevelopmental disorders, but has a focus on autism, examining basic biology to best outcomes pathways. POND, I mentioned before, OBI has made a significant investment over the last 5 years in the province of Ontario to understand how autism fits within other neurodevelopmental disorders and how one can translate molecular biology to novel treatments. Pathways, long Canadian network looking at how our kids grow and how they change as they grow. And Quebec is a site for the Pathways study. There is also a family day for Pathways on Sunday, I think, so some of you may be there. Childbright is the largest recent commitment from CHR to a network that, again, looks across neurodevelopmental conditions, but has a unique focus also within ASD. And then I don't want to miss mentioning the Autism Research Training Network, which is a network that has trained hundreds of researchers and professionals across Canada over the years, across multiple disciplines, was funded by CHR, is looking for a new funder right now. And my last one on this one is I wanted to also show you that, again, all of these are examples that the Canadian researchers and the Canadian networks that you are part of or you're contributing to are also part of international consortia. So we talked about missing before, about the International Genomics Consortium that Steve is gonna talk about. There's also the Autism Sequencing Consortium that lots of Canadian sites are contributing to. Autism Treatment Network, so Lonnie Zwangenbaum from Edmonton and I have the privilege of being of this international network, part of this international network that looks at medical standards for kids, for the care of kids with and youth with ASD, the concept of medical homes and minimal procedures required to facilitate best outcomes. Baby siblings, so international consortium, but there is a Canadian component to the international consortium that looks at how little babies who are born to families who already have a kid with autism go on to either develop a neurodevelopmental condition or not, and what is it that we can learn from that. And the last one I wanted to mention is we have now a formal collaboration between POND, the Ontario Consortium, and EU AIMS, the European Consortium on Autism, where we share data, we work to find biomarkers, and we run clinical trials together of novel treatments for ASD. So all of this for Canada, I'm bringing it back to today. This is our agenda, again, highlighting some of our international speakers in the morning and focusing on doing workshop work with your Quebec leaders in the afternoon and closing the day with reverse performance. Before I'm going to pass this on in a second to Dr. Mayada Elseba, who is my co-chair for the local meeting and somebody who's very familiar to you. So before I pass the microphone to her, I want to thank her and the Quebec group, advisory group, who helped us think through what this day should look like, as well as Dr. F. Marie Quinte, I'm not sure if she's in the room, who is chairing the advisory group. And then I want to thank our sponsors. So this is an event that we fundraise for independently from the same meeting. So INSAR and Azrieli Foundation are our main sponsors for the meeting, but you'll see we had many other sponsors from the Ontario Brain Institute, to Autism Speaks, to Holland Bloorview, to the Miriam Foundation, to your réseau that I cannot pronounce because French is not my strength, to your TAC network, who made this meeting possible. One last housekeeping thing before I pass it to Mayada. For those of you who are using translation headphones, hopefully you have figured this out already. Can you put your hands up if you're doing okay? If you're doing okay, you must be hearing my translation. Okay, good, excellent. So the speakers will have a set of headphones if you need to ask questions in French that will be simultaneously translated to the speaker so they can give you your answer back. And we have Francophone communities logging in remotely also, so we'll use the same procedure. So with that, I'm going to ask Dr. Elseba to come up.
Thank you, Avdukia. Another housekeeping item is to just let you know that the people standing here will not be seeing other slides except if they turn backwards. So if you get a lot of this, don't worry about it. And it's very hard to see the audience, so please give them some good cheers at the end of their talks and keep the day very engaged. It's my pleasure to extend a warm welcome to all of you as well this morning. A special welcome to those who are national and international guests. You've traveled, some of you, very far to be with us today. I'm sure that beneath all the construction that you've seen, the orange cones and the aftermath of floods in Montreal this week, that you still are getting to appreciate what a wonderful city we have here. We actually had the great privilege yesterday to visit City Hall, where we talked with the mayor's team, Mayor Valérie Plante, who was herself dealing with some of the aftermath of the floods, but we were still stayed on the books to be able to discuss with her team how to make Montreal a more inclusive city. So this sort of kickoff to the event was quite successful because we get their commitment to listen and interact with us as researchers and with the wider community that we represent. This outreach to the mayor actually was the first of a series of events that I hope many of you will also attend this week. So in addition to this wonderful day that will be full of knowledge sharing, we're organizing Montreal's first Autism Festival, which is at the intersection of science and society. This series of events celebrates creativity in all its forms, whether it's science, music, arts. We also want to share some of the many successes that we're proud of and we have achieved here in this community while actively thinking ahead on where to go next. And if we want to become an inclusive society, we're going to need a lot more problem solving going forward, and maybe one day we're going to try to lead the way as well. So today is also another reason to celebrate because of the, as Avdukia mentioned, the engagement that we had leading into this day and the festival as a whole. Without a tremendous number of people who have been involved in the organizing and the contributions in many forms today, and none of the events this week could have happened in their current form. We had a very proactive advisory group that was actually convened by an emerging network in Quebec called the Transforming Autism Care Consortium, and I invite you to learn more about the network by visiting our website and actually coming to some of the events and speaking to some of my colleagues in the room as well. This network brings together over 50 researchers across 7 universities here in Quebec. We're very fortunate to be working in partnership with multiple contributing, supporting organizations with funding now reaching in excess of $30 million, which we hope will be put to fantastic use in the next few years. We've been really blown away by the amount of commitment that this group has displayed in terms of suggesting ideas, working through what we can carefully refer to as the tensions that are going on right now between science and the rest of the community, and what the INSAR community as well will try to tackle in the next few days. The afternoon represents a selection of topics that, judging by the numbers today, had also been very popular because they were actually selected and formulated more or less with these community advisors in areas and questions that are quite important for how knowledge that we have acquired in science can be put to good use in our schools, in our hospitals, in our community settings. A final second welcome and thanks to the supporters, the sponsors of today, as well as the Azrieli Foundation in particular. I want to extend a specific thanks to Mira Puri, who's come with us on this journey and has been very supportive as well as an advisor for both the festival and today. Some of you have also will see today some of the members of this committee, and I invite you to speak with them directly. They've been so kind to share with us how they think about the intersection between autism, science, and society in their own words. So please view their stories and find them today and speak to them. For those of us who've been working behind the scenes, it's also quite clear that some of the contributions that certain people are making can never be described in sufficient enough terms. I do want to point out the enormous amount of work, hard work and major commitment that Dr. Evdokia Anagnusto has contributed to make this event happen and to the conference as a whole. I can truly say that she sets the bar very high for the kind of scientific leadership we want to have in Canada and this community deserves. And on this note, we have a small gift for her, and if you could join me in thanking her for her contributions.
Okay, hello, bonjour. I'm going to try, I'm not sure I'm going to try to use my computer here, but I may stop that experiment shortly after I started. But let's see how it goes. Thank you so much to Evdokia and Mayada for inviting me to present today. It's really a great honor to be in Montreal and give this seminar to the general public. And the title of the presentation is Genetics to Crack the Autism Enigma. And these are the take-home messages from my presentation that I would like you to think about at the start of my presentation. So you'll hear throughout the presentations today, but in particular with respect to genetics, that there's enormous variation in clinical and genetic presentation in autism. So collectively, we see this as a group of rare disorders or conditions that share features involving brain cells or the neurons. And secondly, there's enormous variation in the genetic factors involved in autism. We now know that there's upwards of 100 genes that are considered high-impact contributors to autism spectrum disorder. And the hypothesis, and now there's growing evidence, is that by better understanding these genes and the factors that influence them, it may lead to more precision treatments, medical treatments and interventions for people who have ASD. And I'd like to show this picture on the right-hand side of my slide, a famous painting by Tom Thompson. I tried to find a good spring painting, but actually we're not anywhere in Canada in the spring yet. I think in Toronto, we actually had a little bit of snow yesterday. But this is really the way I see autism, that there's lots of different kinds of trees, and they change in color throughout the year and throughout the lifetime, just as individuals change. And what we're trying to do here is to use genetics to understand what the contributing factors are so we can help in decision-making, in decisions around interventions to change these colors in a particular direction. And that'll be the underlying theme of my presentation today. Okay, so far so good. So these are 4 quotes from the first 2 on the first slide from a good friend of many of ours, Peter Zatmari, from a book he wrote called The Mind Apart. And the first goes like this: "It all depends on the way you see things," said the woman on the other side of the little table in my office. "What can seem like a disability one day can be a talent or a gift on another day." And secondly, this is the quote that struck me from his book: "What is so tragic is that the impairments in social interaction, in communication, and in play strike at the heart of what it means to be a child." And then these are 2 more quotes just to help set up the genetics. "People with autism can see the forest for the tree, but they see the trees in exquisite detail." This is Ute Frith. And most importantly, with respect to genetics, this was a quote from Michael Rutter in his scientific paper in 1977. "Examination of monozygotic twin pairs concordant for autism show enormous clinical heterogeneity even when pairs share exactly the same segregating alleles." And the translation of that in English is that monozygotic twins who have the exact same constitutional genome can have very similar presentation of their autism, or they can actually be quite divergent in the clinical severity and presentations. So that tells us that genes can be involved, but that there's other factors also. But this was really the first contribution that started to let us think about a scientific explanation based on genetics, so that there would be a biological basis to this, what was previously defined and still is defined as a behavioral condition. Now, I think what's very important for my presentation is to also acknowledge that there's many other coexisting mental and what I would call health conditions associated with ASD. And this is data from a study in Ontario a few years back, but it's very similar for other studies from around the world, where you see attention deficit disorder, sleep disorders, learning disabilities, depression, anxiety, what I've grouped in red as the mental health contributors, to things like digestive system disorders or brain spinal epilepsy seizures in the black text or in the so-called health conditions. So when we think about the genes that are involved, as I show you, not only is the complexity in how the brain is functioning behaviorally, but also other attributes going on in the body. And I'll try to provide some explanation for this throughout my presentation. Okay, so this is my Biology 101 that I taught my kids who are teenagers now. Why is genetics important? Well, genetics is important. It's your nature. And we'll argue about this throughout the meeting, how much of you are nature and nurture. I think at the end of the day, it's probably 50-50. That's my prediction, black and white, yin and yang. But you can't argue that DNA is the blueprint for your life. And blueprint is a key word because it's a code, it's a blueprint of instructions that give you the instruction book for how you develop, but it doesn't mean that it's absolute. It means that you can actually, in some cases, you can change it by the decisions you make or the way you modify your lifestyle, for example. Okay, so you start out as a sperm and an egg. It fertilizes as a single cell. It doubles, and then it goes to 4, 8, etc. And a typical human being has, as I'll show you in a moment, something along the order of 60 trillion cells in their body. And each cell, other than the red blood cells, have the exact same DNA complement. It may change slightly over time, but it's pretty darn consistent. And the beauty of genetics is that we all share 99% of our DNA. Okay, so in fact, if you compare the DNA sequences from any two individuals here, while different variants are included, the total genetic variation between an individual who's black and white or yellow, for example, is essentially the same. There's just different classes of variants that are involved. Okay, so genetics is what ties us together. It's our common humanity. And by studying genetics, we think we can understand some of the factors that contribute to ASD. And this is just another way to show this. I went back to my childhood. I'm a devoted Montreal Canadian fan. Anybody know who this hockey player is here? You're going to age yourself. I think someone said Rocket Richard, right? Yes, good, thank you. He's actually much older than I, but he's the greatest hockey player of all time. But anyways, I think maybe he had the perfect genome. Well, let's see. But so each of the cells, I mentioned the 46 chromosomes. So these are the packages of DNA. You inherit 23 pairs from your mom and 23 pairs from your dad. And I'll talk about the X and Y chromosome. There's about 30,000 genes that encode proteins strung along the chromosomes. Okay, but really importantly, and we're going to talk a lot about this at the scientific meeting on Friday, only about 1.6% of the overall DNA content codes for those protein coding genes. So there's lots of these regulatory sequences in between. And there's different types of genetic variations, single nucleotide letters or single genetic letters, or these copy number variations where you might have an entire gene or several genes present instead of the typical 2 copies in only 1 copy or 3 copies, or in some cases 0 copies in your genome. Okay, so what's going on in autism? This will give you an answer. So 75% of those 30,000 genes are expressed in the brain. Okay, so the brain is the most important organ in your body. I work at sick kids, we argue with the cardiologists all the time and things like that. But the brain is the most important organ in your body. These genes are turned on. It's like a symphony. They come on at different times during your development. But in the brain, the majority of the genes are on, critical for expressing proteins that are involved in development and maintenance. And as I pointed out, where you have different types of genetic variation, it's very important to maintain a certain level of dosage. Okay, so genes have been selected for and evolved to achieve a certain level of dosage. So there's a certain amount of RNA and protein in each of the cells. And as I'll show you, when you vary away from this dosage, it can lead to differences in how you develop and how your organs develop, how your brain cells work. Okay, so now the technologies to study the genome, and this becomes important because many families are undergoing genetic testing now. This has been happening for a while in the medical genetics situation. For over 50 years, and in fact, some of the technologies to do karyotyping were developed here in Montreal to study the morphology or the outline of the chromosomes, to look at the numbers of units of the chromosomes. This is called karyotyping. And I just indicated it cost roughly, at least at sick kids in Toronto, about $300 to do a karyotyping scan. And then later, after the genome project was finished, there's this technology called Microarrays. And this allows you to scan roughly 1 million DNA sequences across an entire genome. It's on a cover slip of a microscope slide here. And this now is the standard of care test for autism testing because it captures all the chromosome changes essentially, and then much more, the copy number variation in the genome. And it cost roughly the same amount as the karyotype. And I point this out because this test didn't really take over from karyotyping until it became around the same cost. Okay, and now you hear about doing next generation sequencing, one form of which is called exome sequencing. That's where you pull down that 1.6% of the DNA that encodes for proteins and sequence it to look for small sequence letter changes. That's going to become a predominant test in North America very soon. It's already happening more on the research side because the cost is coming down in the same range as karyotyping and microarrays, and it gives you a higher resolution analysis. And ultimately, whole genome sequencing captures everything. Okay, I'll talk about that in a second. And that's about $1,000 for a research genome. If you do it in a clinical lab, it's more expensive. So this is what I think about during the days and nights. This is 6,213 nucleotides from the X chromosome. And I'll refer to this sequence in a few points throughout my presentation. It encodes what we call exon 3 of a non-coding RNA. So this is a new form of a gene that has a function in the cell, but it doesn't make a standard protein, which is kind of neat. And we now know that if a male does not have this exon or this segment of DNA, they have 0 copies because males have an X and a Y, and this is on the X chromosome. They're quite likely to develop autism spectrum disorder, okay? And there's other regions in the genome that encode proteins that also are now known to be involved in ASD susceptibility. Okay, so now I didn't point out earlier that there's actually 6 billion chemical letters of DNA sequence in your genome. Okay, so this is just a very small snapshot of information. Okay, so far I'm doing pretty good, I think. Let's see if I can keep it up. So the evolution of genetic testing technologies, you can have these sequence level changes called gene variants, and we also call them mutations. If it's a genetic change that leads to a clinical outcome, we call that a genetic mutation or a genetic variant. Karyotyping, I mentioned, is already being used, and we see that in, I'm just going to click through this so we have all the information here. This is going to be tricky. Okay, we see that in about 2 to 3% of ASD, we see gross chromosomal changes, and then microarray, we see changes at the level of the page if you think of a book. Okay, so a page can be ripped out, it would be a deletion in roughly 7% of autism spectrum disorder. And for exome sequencing and the newer technology, we see these single letter changes, say in this case changing the context of how a sentence would read. So in this case, the genetic change leads to a single nucleotide change, changing the word beef to beet. And then the sentence still is okay, but it doesn't actually read perfectly. And we see these in also roughly 7% of individuals that have ASD. So these tests are actually happening. In Canada, they're covered at the provincial level. We'll talk more about research at this meeting, but now these types of genetic tests are really rolling out across certainly North America and Europe. Okay, this is a very technical slide. I'm going to show it and really spend a lot of my talk on Friday at the scientific meeting going over the different genetic technologies, but in particular talking about whole genome sequencing. And the take-home message here is that when you use whole genome sequencing in a single experiment that you can do in a few days now, you can capture essentially all of the genetic variants in a genome. Okay, so our group with Autism Speaks, and then I'll talk about others also later too, decided a few years back to embark on a huge project to capture whole genome sequences on thousands of families with ASD so people could use that for their research endeavors. So the message really is if you're going in for genetic testing or you're consulting, you need to know what technology is available to you, what forms of genetic variation it can detect, and what that actually means in the translation, because you'll find some things with one technology and miss some things with other technologies. I do think in 5 years, whole genome sequencing will be the standard clinical norm. It's just not there yet because of the pricing issue. Okay, and as I said, genetics really is the great equalizer, and this is really an incredible slide. This is the experiment I dreamed about doing when I was a graduate student, figuring out how many new genetic changes or mutations that each of us carry in our genome compared to our parents. So this is what we call a pedigree, P1, parent 2, parent 1, parent 2, and then F1 is myself. These are actually my new mutation rates. So I have 73 spontaneous genetic changes or mutations in my genome compared to my biological parents. It's not a lot. It's quite incredible how high fidelity the replication of this massive amount of genetic information is. And these break out into about 60 single letter changes and 13 larger copy number variant type changes. And then throughout your lifetime, you also accumulate some somatic changes. Okay, so we think that roughly 3 somatic changes in the genic regions of the genome are collected typically in a genome. The important point here is that we all have these genetic changes, all of us. This is an average genome would look like this. Some a little bit higher, some a little bit lower. And the changes are typically random in the population, okay? And if one of these changes leads to, say, instead of having 2 copies to 1 copy or 2 to 3, and it hits one of these important neurodevelopmental genes, it can lead to ASD. It's as simple as that. If it's a gene involved in heart development, it can lead to a cardiac anomaly. So we use this approach in our genetic experiments to pinpoint, to highlight these genes in the genome for further research studies. And then the additional evidence I kind of alluded to earlier of the genomic enlightenment in ASD came from the identical twin studies. If you look at identical twins that have the same genome and compare them to the rates of autism in dizygotic twins that share 50% of their genomes, and then also the general population, there's a significant difference. There's a much higher correlation in individuals who have ASD who share their genomic sequences or their genes. When you look in families, there's also a much higher rate, 18% chance for siblings in a family that already has one diagnosis to develop ASD. And we now know these rare copy number variants can be involved, and single gene genetic changes or mutations can also be involved in ASD. There's a lot of talk at this workshop, at the workshops at this meeting, and there's been a lot in the literature. There's roughly 108 different genes or regions that I would consider to be high impact regions contributing to ASD. Probably they're sufficient to cause ASD in that individual, and I'll talk about those in a moment. Okay, so this is the general model. I think the field is adopting of what the genetic architecture looks in autism. There are these high-impact genetic changes, so you might have a copy number variant in one of these critical neurodevelopmental genes that can tip the balance in the direction of ASD. And then there's other genes that are more intermediate in their contributory effect, and there's lots of common genetic variants that are present in all of the population, but at a higher frequency in families with ASD. So they can contribute to the likelihood someone would be on the autism spectrum or pass on their genes. Okay, so the idea really in using genetics is to figure out what individual or what family carries which high-impact changes and also which common genetic changes, and figure out the likelihood combinations of having these and how it plays out in that form of ASD. And then based on this, you might be able to tip the balance back in the direction you want. But to understand really what the best path forward is, you need to know what the balance looks like. Okay, so you're thinking back to the tree analogy. This is actually the way I look at neurons, how they line up. It's a beautiful picture looking at a tree going up the trunk, and it's the same actually with the neurons in the brain cells. Okay, so to understand autism, we have to understand the genetics and the proteins and the RNA molecules that they encode to regulate how these tree branches and trees essentially develop. Okay. So I mentioned there's lots of genes identified now, and this is one of the genes that makes a protein called SCN2A, and it involves a protein that's involved in, we know, in some forms of epilepsy. And it's a protein that is embedded in the cell membrane, and it translates signals back and forth between both sides of the cell. And I just wanted to point out here that the complexity at the neuronal level is incredible, even more so than the genetic level. So there's over 100 billion neurons in your brain, and if you look at the connections or the synapses, there's trillions of these. So there's actually, in my opinion, when you have a genetic change, say a copy number variant or on that little sequence on the X chromosome, and there's any specificity of clinical outcome, it's really amazing to me. But there is. So what we have to understand is first the genetics, and then the effect they have on the proteins, and then the effect on the neurons and the brain as a whole. And you'll hear from Jason and others today about how we do those experiments. But the important point is the proteins that have been identified so far to be involved in ASD are involved in brain development and function. Okay, so this is 4 different models of what the neuron looks like in ASD, and this is what I would call a neurotypical neuron, where in this individual they have 2 copies of the Norexin 1 gene, 2 copies of the SHANK2 gene, and it's a male and has 1 copy of that little sequence I showed you earlier from the X chromosome. So the balance is balanced, it's not tipped in any direction, and there's excitation and inhibition flow going back across the brain cells. Now, in this case, the individual has only 1 copy of the Norexin gene, so there's a copy number variant that leads to less protein being made, so typically it would be 50% of the protein, and it leads to a tipping of the balance, in this case, in the right direction, to the right-hand side. So that's the form of ASD that this person has, at least how their neurons are affected. And in this case, in a paper published recently from James Ellis, Mike Salter, and our group in Toronto, we found that having only 1 copy of the SHANK2 gene, it's also a very important gene that encodes proteins involved in synaptic scaffolding, you see an overbranching of the so-called dendritic branches and the dendritic spines along the neurons. So it's not an underbranching, it's an overbranching. That's a different form of neuronal, what I call autism. So if you were to think about a treatment in this case, the balance tips now to the left direction, you'd want to tip it back to the right. So you have to understand the genetics if you're going to think about a path forward. And then, just as always is the case in autism, it's more complicated than you would think. It's not always underbranching or overbranching of these tree branches or dendritic branches. In this case, it stays the same. It looks a lot like the neurotypical, but when we look closely at the electrophysiology, this is that X chromosome change I talked about where the boy has only 1 copy. It stays the same, but there's difficulties in the excitation inhibition balance going across the neurons, how the neurons actually talk to each other. But they look the same. So in this case, it's a different type of autism too. So that's how we use genetics, and we use other experiments. You're going to hear more about this throughout the presentations today. Okay, this was a nice review written in the Spectrum article just a while back. It says we need to have much more genetic diversity in the databases that we study. Most of the families that you'll hear about that we've studied in genetics are from European ancestry. That's a genetic term we use, our Caucasian families. But to really understand the impact of these rare genetic variants, we need to capture all populations across the world. And as the sequencing costs drop, you're going to see this happening both in the clinical and the research setting to add a lot more power to interpret what the impact of these genetic changes actually is. But this is something we're going to talk a lot about at the meeting this week. And then ultimately the idea, as I alluded to earlier, is to group the different individuals into different categories or groupings based on their genetic changes, based on their neuronal changes, their behavioral changes, and come up with specific information that can inform on diagnosis and on treatment. That's what precision medicine is. So about 5 years ago, with Autism Speaks and Sick Kids, and then also Verily Google, we launched this project to do whole genome sequencing on thousands of families. We decided back then that it was time to dive in and just do the ultimate genetic experiment and sequence all of the DNA. We've now, I think it was announced probably today or it's happening this week, that we've now completed 10,000 whole genome sequences. And the idea is to pay for this once, do it using a standardized technology, and put the data into a cloud. It's a lot of data, huge amount of data. It has to be in a cloud database, and make it accessible to scientists from around the world so they can take their question around genetics to a massive, really expensive, deep database. The Simons Foundation has done a similar project, and there's other smaller projects going on. I'll talk about these in my talk on Friday. But I'm leading the one, it's called Missing, and now we have about 180 different research users across 14 countries taking their questions around genetics to the cloud. So it's really amazing that this is a form of open science that we didn't have access to just even a few years ago. Being in Canada, I have to show the research participation across the country. It's really quite incredible. We've now sequenced close to 8,000 genomes from individuals from families with ASD from across coast to coast to coast in Canada. Okay, 10 minutes. I'll speed up a bit. The iTarget project led in BC by Susanna Lewis, and then the TAC project led by Mayada Elsabag, and then Evdokia leads the Ontario-based project. But we really do go across the country, and all of the names on the slide will either be presenting at the meeting or involved in the meeting at some point this week. Now, we actually mapped the location of these families based on their postal codes across Canada. We have this level of access to information as the research consented for this study. And really the reason for doing this is to better enable the clinical diagnosis. I'll skip through this a little bit quickly, but this was a family that went through 8 different clinics at our hospital in Toronto and did not get a proper diagnosis, not only of their ASD, but also the medical complications that came along with it. And it wasn't until they went into at the time it was a microarray study, now we do this all based on whole genome sequencing, where they found that this individual has a chromosome 16 deletion. Okay, so this was the genetic cause of the autism in this boy. And as I said earlier now, these clinical microarrays, Sick Kids is running, in Ontario, there's roughly 7,000 microarrays run each year, and in Montreal, it's roughly equivalent based on population proportional. Genome sequencing is coming, and the number of results is increasing because you have a higher resolution of genetic detection. Okay, very quickly, I'm going to just go through 3 families here that have got information back, where the index case is shown on the left-hand side, and we found a genetic change in the neuroligin X gene on the X chromosome in the first child who has full ASD, so it's shaded in black. And then the second boy in the family had a suspected diagnosis, and we found the same genetic change very early on, indicating that it was likely that he would develop a form of ASD. So alerting the family and the doctors that there should be behavioral intervention much earlier on. So this was the first time in our studies we've used what I call gene-guided behavioral interventions in ASD. This came from the whole genome sequencing study. In the second family, the 2 ASD-affected boys carried different genetic changes, and the one on the left has a de novo genetic change in the thyroid hormone receptor alpha gene, and the boy on the right has an inherited change from mom in another one of these epilepsy genes that I talked about earlier. In case of the boy on the left-hand side, we've now treated him with thyroxine. I won't have time to go into the details, but there's been, based on mouse model studies, other individuals that have genetic changes in this gene, and this was the first treatment using thyroxine in ASD based on our research cohort. And then on the right-hand slide, this is the very first case we had. This is new unpublished data. We've identified one of those X chromosome deletions in a boy. In 99% of all other cases, it's passed from a carrier mother to an affected boy. Again, the X chromosome scenario. And this encodes, as I mentioned earlier, a non-coding RNA. So this was the first, I would say, bona fide non-coding RNA or non-protein coding gene involved in ASD. This is really important because this is a de novo change, a spontaneous change. So the likelihood that this family will develop ASD, again, based on this genetic change, is essentially the population average, or it's essentially much lower than population, it's 0 for this form of ASD. So that's very important information to have when we think about genetic counseling. Okay, the X chromosome changes, alterations are very informative for these types of genetic counseling. Okay, so the questions that we've answered and that are arising based on this research, why are there so many genes involved in ASD? We're going to talk a lot about this at the meeting, but it comes back to what I showed you earlier. Lots of genes make proteins involved in brain cells, okay? Secondly, are all AS genes proteins somehow involved in regulating and maintaining neuronal synapse development and plasticity? So far that suggests that's the case, but we're only explaining perhaps 20% of the families with autism. There's a lot more going on. So that's a big question in the field. How do we marry advances in genetics and other technologies such as imaging, artificial intelligence, etc.? You're going to hear a lot about that in the next talks. But ultimately, it's to advance the opportunities for individuals with ASD. And I will just show this slide very quickly. This is a new technology we're using where we can take skin cells or blood cells or even cells from urine and to reprogram them into what we call pluripotent stem cells. This is what's shown in the second panel here. And then you can turn these pluripotent stem cells into any type of tissue in the body, including brain cells. So one of the challenges in studying autism is it's very hard to access the individual's brain. I think we can use imaging studies, but to get biological sample, this technology now allows us to do that. So we've now made personalized neurons from some of these families that have these deletions on the X chromosome of this non-coding gene I showed you at the start of my talk. And we can use this to test novel medicines and novel alterations and treatments to see how it affects the neuron behavior and structure. So lots of new technologies, lots of action in the research side. This is going to come in the next talk, so I won't talk about the clinical trials based on the genes. And then finally, it's so important to do this research. This is my last slide. and this is a very unique family, a 4-generation family from primarily Ontario, where we found their genetic change. It's a deletion, a copy number variant in the SHANK1 gene on chromosome 19. And the astute geneticists in the crowd will see that only the male carriers of this deletion, I only have one copy of this gene, develop ASD. The female carriers, even though it's not on the X chromosome, chromosome 19, are protected in some way. These are the key families that give us clues towards new treatments. Okay, so if we can figure out why the female carriers here are protected from the ASD, it may give us clues into new biology and new treatments. So we've now worked with a group in the US to make induced pluripotent stem cell neurons, and we're studying these families and others very, very carefully. So it's really, really critical to worldwide research to have these key families enrolled in genetic research. And we thank all of you in the crowd who not only work on this, but the families who are involved in our genetic studies. And then I'll just leave this slide up. It's very busy, but this is the genetic, the study in the TACC study in Quebec that Mayada and Guy Rouleau and others are leading here. And they have posters and things. All of the genetic studies that are going on here are part of the larger Missing project, and we collaborate and exchange information and technologies and things to move autism research in Canada and worldwide forward. And I'll just close with that and have questions. Thank you.
Yeah, okay, great. So we have about 10 minutes for questions. So feel free to just come up to the mics and ask your question for Dr. Sir. Go for it, Naomi.
Yes. Hi, everyone. I have a question about the types of ASD at the genetic level that you described, and whether that translates directly into the manifestation, behaviorally, language, cognitive in the person with ASD.
Right, so there's been, I say, a growing number of studies are trying to dissect that question. The limitations in the studies right now are still in the numbers that we have because of the vast diversity of genetic changes. So I didn't point it out here very clearly, but thank you for prompting me. None of these genes none of these genes that I've talked about are present in more than 1% of families that have ASD. In fact, the chromosome 16 change I talked about in the Canadian cohort is about 0.5% of the population. Okay, so, and then that X chromosome change is about 0.1%. Okay. So these things do add up to roughly 20% in the hundreds of forms we have. So it is difficult to do these type of what we call genotype-phenotype correlations, but we're trying to. It's interesting, some of the genes, like the CHD8 gene is one that you hear a lot about at the meeting here, don't seem to always correlate with things like cognition, intellectual disability. Some of the genes seem to associate with speech and language disorder, but there's no definitive data yet. What we do as biologists is we group the genetic pathways. So we group the genes that are involved in these individuals into groupings, how they interact in the cell. And I usually show a bubble diagram where it shows how these genes are grouping. And we try to correlate those with the phenotypes or the clinical presentations. And that guides a lot of the drug design and the clinical trial work going forward. And I think you'll hear more about this a little bit later. There are some more definitive examples that have been found, and we published on this, of drugs that you may or may not want to take based on your genetic change, but it's to impact the other complications that come with ASD, not the features of ASD itself, as far as I'm aware. Again, we'll talk more about that later probably. But that's ultimately what we'd like to do, is to have the fine detail correlation of outcomes in several of the syndromes that are ASD related. I think there's over 100 different syndromes that have been identified that carry different names that can present also an ASD presentation. There's much more specific genetic information that can be given. So I didn't have those slides in my talk today, but in roughly 10% of the families coming from our genetic study, the whole genome sequencing study, very specific clinical genetic information goes back to the families based on the genetic change they have. But that's the goal.
Yes. I see somebody standing by the next mic.
I have a question about the research stuff that's going on. We're a First Nations family that's outside of London, actually, so it's close to Toronto. And one thing we're finding is that a lot of this information isn't getting to our First Peoples. I was interested to know, are some of these studies going to be more focused on First Nations people, especially because our numbers right now are 1 in 14 children affected by ASD. So I was sent here and asked on some of this research, is some of that going to be targeting our First Nations children, so some of us can get some answers when it comes to this kind of information?
Right, that's a great question. As I pointed out, we really respect and honor diversity. In our studies in Canada, as far as I know, we've never said no to a family who wants to enroll in our genetic studies. We always find the resources to do it. But there is this challenge of getting people to the studies. Certainly, and I think Myotic can talk about this in Quebec, and I'm sure their study is trying to address this here. In BC, it's the same. In Ontario, our Ontario Brain Institute study, we will go out and get families. We can sometimes bring them to Toronto or to London. We can do that. So, but you're right, we have to be much better at getting the message out to invite people to enroll in the studies.
Yes.
We're trying to do that. We have workshops and things, but obviously we need to do a better job. We would love to have many more First Nations people enrolling, absolutely. To understand the genetics, we need to have good representation of all of those genes across humanity.
Well, and it's interesting too, because for like parents like me, I have 2 on the spectrum, 1 boy, 1 girl. And like a lot of this stuff, we aren't getting the information. We have no idea that this stuff is going on. We'd be full willing participants, but for some reason there's a lack of communication with our own communities. So a lot of us don't even know about the information that's being given or the studies being done. So it'd be nice if we could network a little bit more and get these families involved, because in services altogether, there's this big gap in it.
Yeah, so please come and talk to us after, and then we welcome you to take the message back to the community, and we'll try to help out as much as we can.
Thank you.
We have time, a little bit more time for questions. Any more questions for Dr. Sher? Yes.
Hello. I am no biologist in any way, but one question that I guess it's more so a curiosity, and because I was working with ASD populations last semester in my practicum with music therapy, and it was interesting to see how many more males there were to females with ASD. And I was curious as to why that was. And in your presentation today, I kind of came, it's not a conclusion, but in regards to genetics, since males have the X and the Y and they only have the one X, I'm curious if you had questioned as to why females are protected. And I'm curious if it was due to females having 2 X chromosomes instead of the 1.
Yeah.
So they have more of a chance at being protected because they got the 2. But that's my curiosity. And then my second question is in regards to funding and that how it's being affected in Ontario. Because I know that Ontario, and I could be ignorant to this, but I know in Ontario that there have been pullbacks in funding and things like that. And I'm curious as to how that affects your research.
So the first question, you're a better biologist than you think because you're bang on in here. So a lot of these genes that are involved in cognition are on the X chromosome, and it helps explain some of the preponderance, why we see ASD in males versus females. The biology in males as females is just different. I think if you think of this threshold or balance model, males are more poised towards being behaviorally on the autism spectrum. So if you have one of these genetic changes that's a high impact or a medium impact, males get pushed, the balance gets tipped a little bit easier than females. We have to understand the biology behind that. That's what we're working on all of these projects. And there are some clues that are coming. Hormones are involved, how the brain develops. Some of these genes in males and females they're turned on at different times during the developmental cycle. So there's lots of different answers, but I guess the common answer is there's never only one answer. With respect to the funding scenarios in Ontario, for research, typically in Canada, research is not funded at the provincial level. I think we're very fortunate in Ontario and in British Columbia and Quebec and a few other places where they do get some support provincially for research. I think it's really critical for us to petition the federal funding agencies to continue the investments in autism research. Evdokia presented some numbers there. I don't know, I'd like to see them, how consistent they've been across the years. We need to have more funding to move the research discoveries and advances into the clinic. So I think that's key. And that's what we'll talk a lot about actually again at this meeting this week.
Any other questions? Lovely. Thank you very much, Dr. Scherer. I'm going to invite Dr. Jason Lurch to take the podium.
Oh, bright lights. Hello, everybody, and thank you to Evdokia and Mayada for the invitation. It's a great pleasure to be here. And I'll talk about the question that's been bugging us for a while is there one autism or how many autisms are there? And for those who want the short version of the talk, I don't know. I'm not going to give you an answer either. But I'll walk a bit through the process that we have of thinking about this problem at this point in time and trying to understand where the questions are, where the hints are in the data that we have. So let's start with a little bit of a history of the autism diagnosis. So if you go back 100 years, there was no such thing as autism, there was schizophrenia. And autism, or what we would now call autism, was still considered under the umbrella of schizophrenia. And then as psychiatry and neurology advanced and started to change the diagnoses, it began to divide up schizophrenia into childhood schizophrenia and adult schizophrenia. And this is around the start of the DSM-2, or the Diagnostic and Statistical Manual. So this is really the Bible of psychiatry, and one of the ongoing efforts over the 20th century of moving psychiatry into what at least psychiatrists want to be a true science and trying to find quantitative criteria for the diagnostic criteria that we had. So it began to split into childhood schizophrenia and adult schizophrenia, and then around the Diagnostic and Statistical Manual version 3, that split up into what was at that point called infantile autism. There was a still remaining version of childhood onset schizophrenia, or COS as I called it there, and then adult schizophrenia stayed around. Of course, these proportions are not in true proportions. Adult schizophrenia was much more common at that point in time than infantile autism. That was revised yet again when infantile autism was split up into the commonly known categories from the 4th version of the Diagnostic and Statistical Manual, where you had Rett syndrome, a rare X-linked disorder. You had Asperger's syndrome, autism, pervasive developmental disorders not otherwise specified, and so on. So we're beginning to think that there's no one autism. There's a series of pervasive developmental disorders which can be subdivided into smaller components that are somewhat unique and that are a different category of autism. And again, the division between autism and Asperger's is the best known of those. But then more recently, when the 5th version of the manual came out, that went away again, and those subcategories of autism and Asperger's were reunified into what at that point is now called the Autism Spectrum diagnosis disorder. And the main reason was that it was found to be fairly unreliable to diagnose somebody as having Asperger's versus having autism versus having PDD and OS. It depended too much on local context of what the clinician was thinking at that point in time, and we couldn't really find a strong basis in biology. But that inherently has been the flow over the last 100 years of what this diagnosis is. In many ways captures this problem of, is autism schizophrenia? Is schizophrenia in childhood different than schizophrenia in adulthood? Are there multiple autisms? Is there one big spectrum? Where do these divisions lie? So, I mean, does this actually matter? Does it matter what we call it? Of course, every patient is going to be somewhat different. Does it matter if we call them as having a patient with autism or as having a patient with Asperger's or schizophrenia, or PDD, and OS? And I'm going to argue that it does, and Steve already gave some of the hints of why this is. But effectively, first of all, in terms of the way clinical care is delivered, often the name matters. If you've given a certain diagnosis, that might enable you to have services in school or not, depending on what the diagnostic name is. In terms of regulations, if you want to try to license a new drug, you have to go through different regulatory approvals through Health Canada or the FDA in the US, and you often need to go within a certain category so that they know how to deal with that particular request for a new drug, a new licensing. It can build communities. And in some ways, if you start to know that I am a patient with this particular genetic disorder, you will try to find other patients and other families with that same genetic diagnosis and try to compare notes and have hints about what works, what doesn't work, what are some of the things that I can expect as I grow up, etc. And very often, having more of a hint of what is it that you have, apart from this broad diagnosis of autistic spectrum, can help understand the origins of the disorder as well. So there's some quotes here from a Spectrum article, I'll just read it out loud, where it says, one parent, for example, used the result to refute a teacher who believed her son was just arrogant. And this is in the context now of having the genetics known, but I think this applies more generally to subdivide autism itself. Another parent regretted having looked for easy solutions. In this case was chelation therapy and some of the other non-proven therapies that are out there and can sometimes even be dangerous. Saying, if I'd known that, I would never have considered the stuff that I did. So an argument again that some knowledge of what it is that the disorder is at a more detailed level, whether it's genetics or derived through other information, can help guide the way that you engage with the disorder itself, even if we yet do not have precise treatments for it. Also, distinct biology can lead to different types of treatments. And we're going to switch tracks very slightly away from autism here into cancer, where they've done a lot better than we have in the field of autism research. Where if you look at, all these lines represent different types of cancer diagnoses. And in every case, over the last 20, 30, 40 years, we have started to do a lot better at being able to treat and often cure different types of cancers. And to use one particular example, the medulloblastomas, which is a type of brain cancer that's the most common malignant brain cancer in children, which sits at the back of the brain. I don't think I have a way to point up here. I do have a way to point. So it's this big ugly white thing here is one of the tumors. Again, you go back 50, 60 years, you had a tumor in the back of the brain, it had a name, and you would try to treat it with surgery and radiation. Nowadays, what happens is that the tumor is initially dissected with a biopsy. And of course, that's one of the big differences between cancer and autism. In cancer, you can take out a bit of the tumor and see what that tumor is. You know what the site of the lesion is. And then from there, they take this particular cancer and they put it into different molecular techniques. And they can begin to get a much broader picture of what happens based on what the sequencing information tells them about this particular cancer extracted from this one patient. So the details in this complex diagram aren't really important, but this one cancer can now, using different molecular techniques, be divided into 3 or 4 different subtypes, depending on what the signaling is like. And then once you know that, you have a different prognosis. If you have the Wnt signaling, this side over here, the outcome is excellent. The amount of radiation you need is less, the amount of chemotherapy you need is less. If you have a different type, in this non-Wnt signaling pathway, the outcome can be fairly poor. The radiation dose increases. You start trying different, more aggressive therapies in the hope that you will get there. And through this process of having a molecular basis of the different subtypes of cancer, from what used to just be a cancer in the back of the brain, we're starting to get better treatment. So the hope is that as we understand the biology of what the subtypes of autism might be, we can get to either better understanding and eventually better treatment of these different subtypes. Okay, why is this all so complicated? So why do we have this splitting and merging of different diagnostic criteria over the years and still don't really have a good sense of where these boundaries lie? So some of this is simply that in the classic quote, once you've met one person with autism, you've met one person with autism, is that everybody presents somewhat differently. So within what we call the autistic spectrum, you might have somebody who has strong language abilities or who's nonverbal, might have seizures, might not have seizures. If you go through the different comorbid diagnoses, 70 to 80% of people with autism can also have a diagnosis of attention deficit disorder, but 15-20% can have a comorbid diagnosis of obsessive compulsive disorder. Many of them have a comorbid diagnosis of some kind of anxiety disorders, depression, but not all. So one person with epilepsy, other person without seizures might present quite differently in terms of some of the clinical needs. So it's a very complicated, very heterogeneous disorder. So related question then is, what are the basis of the biology of these different diagnoses? And it's another way of saying that if we're confident that we have the diagnosis right, then there should be a core element that's very much shared in terms of the underlying biology and not just the symptom presentation of patients with autism. So I'll go a bit over into genetics again, and this really is work very much inspired by Steve Shearer and his group, and I'm, unbeknownst to Steve, stealing a lot of his slides. But we're very much in the game in autism of having these rare genes. So really what this means is that if you switch tracks to different diseases, look for example at Alzheimer's disease, one of the aging-related dementias, most of the genetics that we know about are risk genes, which are common, which means that, for example, the ApoE gene, about 15-20% of the population have a certain variant. If you have that variant, your risk of getting Alzheimer's increases 2 or 3-fold. In autism, it tends to be very different, where it might only be 0.5% or at most 1% of the population that has this particular variant, but the risk increases many orders of magnitude if you do have that particular variant. And then that means that rather than having a few genes each providing some amount of risk that we know about, we have many, many genes, each of which only ascribe to a certain small part of the population. So the individual genes that are on this list here, again from another slide stolen from Steve, don't particularly matter. They're not very poetic. Names like CHD8 and ARID1B don't really roll off the tongue. But there's many of them. And then the question becomes, we're thinking of autistic diagnosis. Does every one of these genes really represent a separate disorder? Are you an entirely different patient, whether you're a CHD8 mutation carrying patient or an ARID1B carrying mutation? So are we switching from having the different autisms subdivided into one autistic spectrum? We go to genetics, and we now have many of these different individual disorders, each of which are one part of autism that are now distinct and have to be treated separately. So there's a lot of potential power here, and there's a lot of worry in the community that if this turns out to be true, if this turns out to be the case, we're in quite a bit of trouble in some ways. Because for many of these genes, you might only, within Canada, have 5, 6, 7 patients who are known carriers of it. You have hundreds of them, if each of them a different disorder, trying to find different therapies for them is going to be a difficult task. And at this point, if you were to take 100 patients, you'd only have a likely causative gene that you can identify in 20 to 30% of them, leaving the other 70% completely unknown. You can also start moving one step beyond that and say, there's a list of hundreds of individual genes. Maybe the individual genes isn't quite as important as the pathway that this gene lies on. So for example, if some of the text got a bit messed up here. But if you were to imagine grouping these genes as having different types of trees, so you have apple, pear, oranges, rose, and orchid, you could imagine them having grouped into different subcategories. You take those and have the fruit versus the flowers. All of those turn into plants. So somewhere in that grouping might be more information than just saying it's a pear and apple, but you might be able to do something that's generic across all fruit. So if you look at some of this type of data, you come up with a series of different signaling pathways. And there's more than 3 of them out there. We're going to focus on one particular paper from Steve's group, again, came out fairly recently, where they saw that there were really three very core groupings in terms of the genetics of the disorder. And these include the synaptic component, the transcriptional regulation, and RNA processing. So I too am now going to go back into a little bit of biology 101 to go over these more overarching groupings. Where if you think of this synaptic and neural adhesion, if you look at your classic neuron, you tend to think of neurons not as individual things, but as big parts of a network of communication. So one neuron communicates to the next one, and this happens in over billions of neurons and trillions of these connections. And so what you have, if you see it here, you have one neuron, it sends this little process out, and it talks to another neuron, and if you zoom in, there's this place here where chemicals are released, and that's what happens in order to control movement, in order to control thinking. Now, for this to work, this is a zoomed-in case, you have a very complex molecular machinery, but something that keeps coming up and up again are these little bits here. So effectively what happens is two neurons have to have their connections close to each other, and they have to make sure those connections stay close. It's almost like shaking hands across the synapse so that the one neuron that's on the one side of it can send signal to other neuron that can be there to receive it. You start messing with that, you still have a viable functioning brain, but all of the signaling, all the communication begins to be slightly distorted and slightly messed up. So the second of these big bubbles is in transcriptional regulation. So again, to follow up on what Steve is saying, when you really think of the idea of going from this DNA signal to the ultimate outcome, you think of switching from DNA into proteins, which is this whole process of going from transcription of the DNA into translation into the amino acids and ultimately the proteins at the end of the day. And this transcription process is fairly complicated. There's multiple different bits of molecular machinery that are needed in order to help that process move along. And the two key ones are the ability to tell it, okay, go translate this particular bit of DNA, or do not translate this particular bit of DNA. And if you start to change these activators and deactivators, you begin to have some issues in terms of having the right protein at the right place and time, and you start to again have slightly fuzzy brain signaling. So again, this kind of activating and repressing. And then the last part of this set of bubble, the RNA processing, goes along the same lines as well, where again, you go from transcription to translation. But there's this part in here where as you go in between them, you have to process the RNA. You have to get rid of the parts that aren't part of protein, assemble the whole bit of RNA segment together, and move it on. And you start to interfere with this part here. You again don't quite optimally go from having DNA into having the protein at the end of the day. So you had three big bubbles, one of them directly related to how neurons communicate, and two of them related to how DNA gets turned into protein, each of which can slightly and subtly mess with the overall intactness of the system. So if you're thinking about it that way, maybe we've now gone from having the 5 different types of pervasive developmental disorders into the overall autistic spectrum, the way that we talk about it now. Maybe those get subdivided into all of the individual genes, and potentially we can follow that up with yet another step, which turns the individual genes into different subgroupings of it, including the ones that are involved in transcription or translation, RNA processing, and the way that neurons talk to each other. Of course, again, this still leaves us with the issue of those where we have no particular genetic diagnosis. We don't know much about where they could be categorized in here, in this particular process. So switching tracks a bit away from genetics, what about the more pure biology? What about the biological phenotype that we can measure? In this case, I'll focus on brain imaging. There's multiple other ways to get at the same kind of questions. Steve was showing some of the cell modeling type strategies that you have. But if you're thinking of brain imaging, it's the best way that we have right now to, in a non-interventional way, begin to look at how the brain is working. So looking at brain structure, so how big is your brain? How big are different subparts of the brain? We know that that bears at least a loose to somewhat strong relation to brain function. It's strongly influenced by genetics and influenced by the environment, the usual interaction of nature and nurture. So if you look at some of the data that's coming out, and this is from a very elegant study done actually here at the Montreal Neurological Institute by Boris Bernhardt and company, they found that certain brain areas are more likely to be different in patients with autism than in otherwise typically developing controls of the same age and sex. And looking at some of our own data, the same thing comes out. So I'm showing side views of the brain here, and anything that's colored areas that are more likely to be abnormal in a patient with autism than in a normally developing control who does not have an autistic diagnosis. But if you look at the individual data, you can see that this is very, very noisy. So this is just one example here, where you have the surface area, so the amount of cortex there is in that case, and all of the patients are in the different blue dots, so these are individual dots, each dot represents one patient. All of the controls, typically developing subjects, are in the red dots. So the odds of being able to diagnose based on this information are very, very close to chance. So it was not diagnostic information at all, it's just once you have a large enough sample, you begin to tease it apart a little bit. So there's something to be understood in this particular variation. And again, to go back to the same work from this group in Montreal, they began to ask, can we take these signatures in terms of brain structure and begin to subdivide them? Are there some patients that happen to have a larger frontal part of the brain and some patients that happen to have a larger posterior or rear part of the brain? And do they begin to group differently? So in fairly clever work, they were able to pull this together and came up with what they considered 3 different subgroupings of autism based on this pure brain structure information. And then if they used that to try to predict the severity as measured by ADOS scores, a particular diagnostic instrument, they found that if they didn't use any subdivision, they just looked at brain structure, all of autism in their patient set versus all of control, the prediction was fairly poor. But if you look at the lower plot, if they subdivided it with their 3 different clusters, they began to get a somewhat better, still by no means perfect, but somewhat better prediction of symptom severity based on knowing what the different brain phenotypes might be. So this is the idea now that rather than going from one overall autistic spectrum, we now use biologically informed criteria, such as these brain images, to begin to subdivide it in this one example that I was showing 3 different subtypes of autism. So to go back to comorbidities, that's only looking at autism itself. Again, as I mentioned earlier, and as Steve mentioned in his talk as well, many patients with autism will also have a diagnosis of attention deficit disorder. Some will have seizures, some will have obsessive compulsive disorders, many will have issues with anxiety and depression. So do those begin to inform us? And maybe more importantly, does that say something about just how distinct is attention deficit disorder, for example, from autism itself? I'm going to go to a Swedish study which very powerfully informed this, which was looking, and one of the things that the Scandinavians have done very, very well is they have very strong public health records. So you can begin to ask questions on almost the entire population based on the public health records that are very difficult to do in North America given the data that we have access to. So they asked in this patient group of 1.8 million subjects, not in patients though, 1.8 million kids, what's the likelihood of having attention deficit disorder if you have autism? Which was informative, and the odds were very much higher that if you have one, you have the other as well. But then they asked the genetic component of that. If you have an identical twin, or if you have a non-identical twin, or simply brother or sister, and you have autism, what's the chance that your twin brother, your twin sister, or your non-twin brother or sister also has ADHD, so not autism? And the plot showed a remarkable genetic trend of these 2 disorders being linked together. So I'll walk you through this. I find this is a little bit tricky to understand, but very, very important to me at least. It's a bit hard to read, but on the top list you have the within individual, and on the axis here you have the odds ratio, which is the odds of having the other disorder. So if you have autism, your odds compared to having ADHD compared to somebody who does not have autism are about 26-fold higher or so. If you have autism and you have an identical twin, the odds of your twin having ADHD, it's not autism but ADHD, is almost 16-fold higher. And this goes down in this very classic pattern that suggests something about genetics, where it's 20-fold higher, 16-fold higher if you have an identical twin. It's down to about 7-fold higher if you have a non-identical twin, which is the same as if you have a sibling. It goes further down as you have cousins, half-cousin, etc. So this pattern, even if we don't know the genes that might say this gives you ADHD, this gives you autism, it clearly shows that the disorders must be very strongly linked if the heritability suggests that having one gives you much higher chance of having the other as well. Looking at some imaging data now, there's a study that Stephanie Mays led out of Toronto. She looked at some of these different neurodevelopmental disorders, or disorders of brain development, and began to ask to what extent are they actually different from each other. So she looked at the signaling pathways in the brain, the white matter, and she compared autism with ADHD, with OCD, with typically developing controls. And then she found there were some brain areas that were universally affected across the three disorders and distinct from controls themselves. And if she went into the different subtypes, so comparing ADHD to ASD, and apologies that the text is slightly cut off here, she found that only some of those ended up being different from each other. So there were some differences between ADHD and OCD, but there were no differences at all in the brain signature of ADHD versus ASD. And overall, most of this related to having the core idea that these disorders, that we've always considered distinct, if you think of having entirely different diagnoses, were actually quite difficult to separate from each other. Somewhat related data, looking now at not the white matter in the brain, but looking at the functioning signaling pathways, so looking at the changes in blood flow as you're sitting and watching a movie, as you're thinking about this talk, as you're trying not to fall asleep while listening to me. You can begin to ask, how do different brain areas relate to each other? How do they fire together? And when Eunjung Choi from Toronto began to look at that, she again found that there were some distinct signatures in terms of how different brain areas communicate, which were completely shared between OCD, ADHD, and ASD as distinct from the control population itself. So the networks were decreased, some were increases in those networks, some were decreases. But the comparison between ADHD and ASD and obsessive compulsive disorder were not different, but the comparison of all of those brain development related disorders towards typically developed controls were significantly noticeably different. So this is this idea that maybe we're not even just talking about having autism, whether it's subdivided or not, but we're talking about a large pile of neurodevelopmental disorders where OCD, obsessive compulsive disorder, and attention deficit disorder aren't even easily separable from autism itself, that there's something that changes how the brain develops and how we grow that is actually quite universal, whether your symptoms are more related to having obsessive disorders, attentional disorders, or social deficits. You can take this and begin to start clustering things. So I showed a little bit of this with the data on the autism population from the Montreal group, but now we can start to throw all of the different diagnostic groups in. So in work from Azadeh Kushki in Toronto, she did this based on brain structure and some functional outcomes, and she came up with 10 different clusters. And what's interesting is if you look at these plots, the bars show how many patients are in each cluster, going from cluster 1 to 10, and the colors correspond to what their primary diagnosis was. And what's clear in looking at this, you're not coming up with one cluster, it's all the patients with autism, and another cluster, it's all the patients with ADHD, and another one, it's all the patients with OCD. You're coming up with a mix. You're coming up with some clusters that are a mix of patients with, here you have lots of typically developing controls, and some patients with OCD, some with ADHD and ASD. Over here, you have a mix of autism and ADHD. But in no case are we getting, from this readout from brain imaging, a pure separation of autism from ADHD from OCD. We're getting a mix. We're getting subgroupings that unite and split apart these different neurodevelopmental disorders. If I go back to the functional data itself that I was showing you just before, how different brain areas communicate with each other, in looking at this data, Eunjung Choi and her colleagues came up with 2 different clusters. And if you look at the numbers between them, in the first cluster you had 60 patients with autism, 23 with ADHD, 8 with OCD, and some controls. In the second one you had autism, ADHD, OCD, and some controls. Again, not clear separation, and even some of the symptoms being able to meld together with patients that are considered typically developing controls. So maybe this is the idea that you have autism that can be as one autistic spectrum, which is really part of a much larger neurodevelopmental disorder set, including obsessive compulsive disorder and ADHD and others, which can in turn now be subdivided into maybe 2 different types of neurodevelopmental disorders containing a mix of autism and ADHD, or maybe 10 different types of neurodevelopmental disorders. Again, a lot of uncertainty still based on us beginning to try to understand the biological signature of these different disorders, and even understanding which signatures in terms of brain structure or genetics, whatever, et cetera, are important. I had previously talked about the different genetics, and maybe they can begin to be tied back into the brain imaging or other related biological phenotypes. And as Steve mentioned in the answer to the question from Dr. Azrieli, that's actually very challenging to do in the human populations because of the way that the genetics is unfolding in autism. So this is this idea that if you're looking at the genetic spectrum, if we're talking about autism and some of the related disorders, we're really in this game of rare variants. So we're coming up with tens and hundreds of genes that are linked to autism, but each individual gene will have a very small number of patients in it. And the biology that we're measuring is noisy. So those genes might influence brain structure, might influence brain function, but so will a lot of other things within the perfect range of normality that will give you a signal that's different to separate out if you only have one particular patient. You have no confidence that this is really something that's about this particular gene that might have caused it versus something that's idiosyncratic for this one patient. So there are a few studies where you can do this. One of the most common mutations is the 16p11 copy number variant, one particular part of the genome, which is linked to autism as well as some other disorders. So there have been some studies, and I'm showing one example here, which have been able to look at those patients in cohorts gathered from across the world and found certain brain areas that are very much affected in those patients themselves and not in patients or not in subjects who not have this particular mutation. But we can do this for maybe 3, 4, 5 different mutations, and we do not have the numbers for the other mutations in the data set. So this is where we've started to go towards the mouse. And the core idea is that the mouse is not just a small little furry human with a tail, but that it's a similar enough species. I mean, 80 million of years of separation of evolution do something, but you still have a brain that's structured in a fairly similar way. You have a genome that's structured in a fairly similar way. And we can begin to test these individual mutations and have sufficient power in the mouse to begin to understand what does this one gene do. So the idea is that we find a gene in the human studies, we find the mutation that's affected, and we create that mutation again in a mutant mouse model. We can then compare that to its own sister or brother who does not have this particular mutation. And we can do this again and again to get sufficient mice. Now we can breed them to get the kind of understanding of what's the difference between these particular mutations. And we do this for one particular gene. We then do some brain imaging or other assessment of biology of what's happening with this mouse. We analyze the data. And if you use one example here, I showed you the human data on the 16p11 mutation. You can do the same thing. You can create this kind of copy number variation in the mouse genome and ask what's happening in the brain. And if we compare the signal between mouse brain and human brain, many of the same areas that we know are influenced if you have a deletion or duplication of 16p11, this one genomic region, are also influenced in the mouse. It's not perfectly identical, but it's enough to give us some confidence that we're talking about similar things if we look at the mouse versus the human. Then, of course, you can begin to do this across not just one of these genes, but all 20, 30, 40, 100 genes that you have and study each of them and begin to ask what's the similarity and what's the difference between all of these individual genes. And that's partly what I'm showing here. So in this plot, effectively every single bar that you see corresponds to one particular mutation where we have sufficient mice that we've bred to understand what does this mutation do. And on the axis here, so whether it's pointing up or down, is telling you whether this particular gene caused the brain to increase in volume into a bigger brain with the mutation, have no change if it's sitting around 0, or caused a decrease in brain volume. So the individual genes, again, the impact of them isn't particularly important for this one plot. But you're already seeing, in this case, 60-some genes, each of which are associated with autism, and you see this large variation. Some cause an increase in brain volume, some cause a decrease in brain volume. They're not all doing the exact same thing. Different genes are pointing towards different outcomes in biology. I'm going to skip this very slightly, and you can take this kind of data and group it together to begin to understand, are there some pathways again that start to have a similar phenotype in terms of how these genes affect the brain? So when we do that, we can come up with multiple of these pathways and say there's 5, 6, 7 different ways that individual genes can influence a particular brain development outcome, which to some extent begins to reflect that early idea of genetics that we had, but now based on measuring the phenotype in the brain, as opposed to just knowing individual things about the gene itself. So maybe we go from having different autisms to having different neurodevelopmental disorders, which can in turn be subdivided into subtypes of neurodevelopmental disorders containing a mix of autism, ADHD, OCD, and other disorders. And maybe those again begin to come up with certain pathways that signal whether it's RNA processing or transcription or neuronal adhesion that's influencing how the brain works. So I'm going to wrap up here. Again, I asked the question of how many autisms are there. I still don't know the answer. But I think it's fairly clear that this concept that we have of an autistic spectrum, it's a useful one because obviously there's something that's shared if you're on the autistic spectrum. But there's so much variation within it that in order to begin to move towards treatment pathways or even better understanding, having an idea of what the prognosis might be, having an idea of what some of the comorbid clinical issues might be, begins to need to subdivide these disorders. If we have to subdivide it into every single gene is an entirely different disorder, it's going to be a difficult challenge because we cannot identify a gene in many subjects. Now, whether it's because we haven't found the gene yet or whether it's because it's a non-genetic reason, we don't know. I'm sure it'll be a mix of the two. But then we end up with not having one autistic spectrum, but having hundreds of them, each of which are very, very rare and very difficult to begin to understand because of their own rarity. So autism is highly heterogeneous. It has a strong overlap, especially in terms of biological signature, with attention deficit disorder, anxiety disorders, seizure disorders, obsessive-compulsive disorders. It's highly heritable, but as I showed from the data from Sweden, is that the risk of having autism also much increases the risk of your related family members having ADHD, and not just having autism itself as well. So it suggests that there's more than one autism, hopefully not one per identified gene, and there's enough evidence that these genes do begin to group together into pathways and that the biology groups into things that aren't just completely separate depending on what the genetic cause might be. It is going to be mixed in with what we now consider the other diagnoses.
Among your research community, is there an acknowledgement of cortical visual impairment as a comorbidity and its effect on behavioral presentation?
So there's certainly an acknowledgement that sensory issues all around, whether it's visual, whether it's tactile, whether it's auditory, are a significant component of autism. And exactly how they fit into it, is there going to be eventually a subgrouping of patients with autism who have a visual abnormality, deficit, whatever it might be as well, or whether that's going to group in with all of the other sensory issues, or whether it's even going to group in and relate to seizures, that we don't know yet. But certainly sensory issues are, I think, a significantly underappreciated component of autism and some of the other neurodevelopmental disorders as well.
Okay, thank you.
Other questions? All right, so I think you answered all questions during the talk, apparently, so thank you very much. We're going to take a 15-minute break if we kindly ask you to come back here in time for the next round of presentations. Going up next, so please help me welcome Dr. Robbins.
Thank you. It's nice to be here. And I want to thank the organizers of today's pre-conference for inviting me to come and speak about my work on early detection of autism spectrum disorder. Just a few disclosures first for you to take a look at. And I also acknowledge my collaborators in much of the work I'll be presenting today. And this is a very outdated photo. It reminds me that I need a new picture, but I'd like to acknowledge my team at the A.J. Drexel Autism Institute for all of the hard work that they do that contributes to the science I'll present today. My objectives for today's presentation are to review the common warning signs for autism in very young children. I think many people are familiar with the presentation of autism in preschoolers, school-age children, and perhaps adults, but looking for those earliest signs during the first few years of life is something that may be less familiar to some people. Then I'll emphasize the importance of early detection. And I want to mention that there is some controversy right now in the world about what the best practices are for early detection. And I'll be mentioning some of the work we do in the United States, where the American Academy of Pediatrics has recommended universal screening for all toddlers during their well-child visits. But I'm aware that in Canada, they have made the determination that there is not yet sufficient evidence to make such a recommendation. But what I'll do is talk about some of the different strategies that we can use to detect autism in young children. And I'll use some of my work with the Modified Checklist for Autism in Toddlers as a model for how people in the community can understand the literature and make decisions about what tools might fit various clinical, educational, or research needs. So I'm sure that people in the room are familiar, but just as a quick reminder, we're looking at two domains of symptoms in ASD, and we're looking for the absence of expected development in toddlers in the social and communication domain. So we're looking for children who are not developing the expected behaviors that engage with other people and use verbal and nonverbal strategies to communicate with others. Less common in toddlers are the early emergence of restricted and repetitive behaviors, but we see them occasionally simultaneously with the lack of social and communication development. At other times, these symptoms might emerge a little bit later as children are aging into some of the symptoms that require higher cognitive or intellectual levels. And again, I'm not sure that there's total concordance across the world, but in the US, our Centers for Disease Control and Prevention do a big prevalence study every 2 years, and their latest numbers are that 1 in 59 individuals has autism. So now what I'd like to do is switch over to show you some videos. What I'd like to do first is illustrate what we see in a typically developing child in a social situation, and then show you another child who is struggling to engage in the activities. And I ask that out of respect for the families who gave me permission to share these videos, but asked me to keep them protected, that you please not record these videos. So now we'll go back to the slides. Great. So many children are showing symptoms before their second birthday, yet the average age of diagnosis is still after the 4th birthday. And this is true in the United States, where they've done rigorous studies of first diagnosis, and also in many other regions around the world. We've also identified that parent concerns prior to diagnosis do relate to children's later diagnoses and their needs for early intervention. So I think 10 years ago, 15 years ago, a lot of medical professionals were cautious about parents because they weren't experts. They didn't have the background in development to know what was typical or not typical. But it turns out that parents are the experts on their own child's behavior. They may not know that their behaviors form a constellation of symptoms that we call autism, but they know that something isn't developing the way they expected it to. And so eliciting concerns from parents is a really important part of the early detection process. I also want to mention that we see significantly increased delays in detection for underrepresented minorities and families who have low income, so low socioeconomic status. And this is a disparity that we need to address and is part of the public health problem in not identifying children with autism, particularly those who may be underserved. So just to take a moment to point you in the direction of why this is the body of my work is about early detection, if you can identify among a group of many, many children, the few children who are at risk for autism, what you can do in the short run is reduce the family's stress. So when parents are worried and they don't know what to do or where to turn for help, it can increase their stress quite a bit. But even more importantly, early detection facilitates early intervention. And although in many parts of the world, children can get intervention without a formal diagnosis, most often that's not the kind of intervention that's at the intensity or the specificity needed when a child does have autism. And there's a growing body of evidence that children who are detected earlier have more positive outcomes across their lifespan. I just realized that I wasn't advancing the slides, and so you've been doing it for me. Thank you. Sorry, I'm a little slow. I'm used to presenting right off my laptop. But the people in the back are doing a great job. I can try to take over. And so just a few more words on the specific kinds of intervention that are warranted when a child does have an autism diagnosis. So I've just mentioned a few of the types of intervention that have the strongest evidence base. And from the early 1980s, there's been a growing body of literature on applied behavior analysis. And much more recently, there have been some newer forms of behavioral and developmental interventions that integrate both behavioral approaches with other aspects of early development. And these include models like the Early Start Denver model and pivotal response training, but there are others. For a long time, as these separate manuals were being produced, the authors were all very certain that their treatment was distinct from the others and that they really didn't do the same thing. And I feel like it was a landmark shift in the field when in 2015, the authors of a number of these different intervention programs came together and realized they actually had more in common than they had previously thought. And so the term was sort of born, I think, around this time that labeled many of these interventions as naturalistic, developmental, behavioral interventions. And it's a mouthful, but the point is that the very earliest behavioral intervention research was lab-focused. It was very restricted. It took a child out of their usual environment. And so shifting to a more naturalistic and developmentally appropriate way of using those behavioral strategies, I think, embodies these newer treatments. So to shift back to the video for one more video, I just want to give you sort of an inspirational video. Not every child who gets early intervention shows this much progress, but this is a little boy named Wells, and the videos show excerpts from when he was about 2.5 and first starting intervention until he's about 5. And watch how in the first clips, it's really hard for the therapist to get his attention, and she has to work very hard. And then you see some clips where he's doing a little better, but he still needs a lot of prompting. And then it jumps to a little more than a year later, and you see his independent, not only speech, but also his engagement with his father in a really fun pretend game.
Beautiful, that's standing up like this, okay? Let's try it again, stand up. That's standing up. Say, juice. Juice.
Yeah.
Nice try. Look at Georgia. Say juice.
Yeah.
Juice.
Yeah.
Good. That was a much better try. Which one? Lollipop. Yeah, where's Georgia? Thank you. What's your name? Say Wells. That was better. What's your name? Wells. How old are you? 2. How old are you? 2. I like it. Do this. Mwah. Beautiful.
Here. Take a picture with you, Carmen. It's upside down.
Picture, okay.
It's upside down.
Okay.
Put
I gotta go on.
walls through here.
All right.
First, you have to take your glasses off.
All right.
Like me.
Okay. Okay, I got you.
Okay.
All right. I got a motion picture camera though, so I can keep it going. I should be filming this. Got a picture of you taking a picture of me.
I took a picture of you laughing.
Okay, good. That's always a good shot when you're laughing.
Maybe I could take a picture of you.
Okay. So you can see that this little boy went from being extremely resistant to looking at the therapist, making any word approximations, to this level of engagement and interaction. He's not completely typically developing at this point. He still has some speech impediment, and we don't know yet what kinds of other challenges he'll face as he gets older. But it's a humongous improvement, and one that we would like to make available to all children. So now what I'd like to do is switch gears and talk about strategies to detect ASD. In the US, a lot of our guidance comes from the American Academy of Pediatrics, and I've given you some quotes from them. They basically recommend a two-pronged approach that involves ongoing developmental surveillance. And if you think of surveillance as the aspect that requires the expertise of a trained professional to integrate information that they're collecting during a clinical visit, that will give you a good sense of surveillance. It includes asking parents what they notice at home, what their child is typically like. It includes looking at prior records from earlier visits to look at change over time, assessing risk factors as well as protective factors, and making clear notes of what's expected in future visits so again they can track progress. So these are very important strategies, but if you use surveillance alone, it's not likely that most of these expert providers will see the earliest warning signs of autism, because not only are they subtle, but many of them are difficult to detect in a well-child checkup. To complement surveillance, there's screening. And screening requires using a standardized tool. And if you're going to use it in the whole population, such as in universal screening, it has to be brief and low cost. And it aids the identification of children at risk for developmental disorder. So I wanted to just point out a few important words in this definition. Standardized means that it's reliable and valid, and we'll come to define those in just a minute. That it's administered the same way to everyone, that it's scored consistently, and that it provides a clear algorithm for how to interpret the results and what to do next. You can see that that's very different than the more qualitative, using your clinical judgment approach in surveillance. I also want to point out the word aids. It aids the identification. You can't just give a screening tool and be done. It has to be integrated with the other information that you gather during a checkup. So screening cannot be done in isolation if you want it to be an effective detection strategy. So it's really intended to complement the surveillance. It's also crucial to realize that screening is not a diagnosis. It only indicates that the next step should be a referral to an expert clinician who can identify whether it's autism, a different kind of developmental concern, or perhaps not a concern at all. So coming back to reliability and validity, reliability is basically how consistent your tool works when you use it in different settings or repeated over time. Validity is the ability to discriminate those children who really are at risk from those who are not. So is the tool really measuring what it's supposed to measure? And so I want to give you a little bit more information on how we measure validity in a test. I'm not expecting you to take down these equations or anything, but it will help you understand when you're reading the literature what these terms mean. So you can consider that every child will have a place in this 2x2 table at the bottom. So if you give a screening tool, the results will either be positive and suggest risk, or negative and suggest not at risk. And there will also be an underlying true diagnostic state. In this case, it's autism or not autism. And so every child can be classified in this type of matrix. And what we hope is that the boxes that are in yellow are most of the children. That if a child really has autism, they'll screen positive and be a true positive identification. And if they really do not have autism, that they'll screen negative and be a true negative identification. But a screening tool cannot be perfect. Remember, if we're going for brief and cost-effective, it can't be the very best discriminating diagnostic tool. It has to be a brief screener to just pull in the group of children who need to be looked at more closely. And so we end up with false positives and false negatives. And so the way we measure how a tool is working is by creating equations using these 4 cells of our matrix. So sensitivity is the ability to detect the autism when it's truly present. And it's basically a formula of the true positives over that top row of all children with autism. And similarly, specificity is the ability to detect not autism or wellness when it's truly present. And so it's an equation of the true negatives over the bottom row of your table. The other way you can look at this is how much faith or confidence you have in the result from the screening tool when you don't know the underlying diagnosis. And so positive predictive power or value is the likelihood that your positive screen result is a true positive and not a false positive. So now it's an equation going down your table. And negative predictive value is your likelihood that your negative screening result is a true negative and not a missed case. And then you can use these formulas to create even more formulas. So things like a likelihood ratio is a formula that looks at sensitivity divided by 1 minus specificity. And there's a suggestion in the literature that if your likelihood ratio is at least 7, your tool will help you above and beyond what other detection strategies offer to find the children who are likely to have autism. So the biggest challenge when you're using a tool, like I'm going to show you in a minute, for universal screening, is that there's always a trade-off between false positives and false negatives. And so in our work, we really tried to minimize the false negatives or the missed cases, thinking that those children are in great need of early intervention, and we wanted to be sure that we caught as many of them as possible. So to do this work, I'll introduce our screening checklist. It's called the Modified Checklist for Autism in Toddlers, and now we have a revised version where we've integrated the second phase of screening, the follow-up, into a single tool. And we first published preliminary findings on this tool back in 2001, and since then it's been adopted and recommended by the American Academy of Pediatrics, the CDC, and a number of nonprofit organizations as well. And not that I expect you to read this, but just to show you that it's a single page, either the original MCHAT or the MCHAT revised, and up close to just show you a little bit more about it. We ask parents to tell us if their child usually does each behavior or not. And if they've only seen it once or twice, to answer no. And then it gives a variety of questions. Is your child interested in other children? And then to give some developmental context, we give some examples of what an 18-month-old should be doing to show interest, watching other children, smiling at them, approaching them. And then if a child shows initial risk on the MCHAT, then we launch into more detailed follow-up for those at-risk items. So most children don't need these follow-up questions, but if a child is showing risk on some items, we get more information. So if a parent has reported that the child isn't usually showing things, we ask some very specific examples, just in case one of them makes the parent say, oh, yes, he loves to do that. in a sense, it's that the parent may not have thought of all of the possible opportunities a child might have had to demonstrate a behavior, and so we ask for more detail. Other times we're asking about frequency or about how the child first approaches a problem and then what they'll do if they're not successful. And so this work has been presented in the journal Pediatrics, first with their original MChat and then with our MChat revised, if you wanted to go and download the scientific papers. And just to take you through, I wish I had a pointer. Is this a pointer? Ah, perfect. So just to take you through a little bit of how we come to the numbers that I'm going to present. This is our total sample here from the MChat revised validation study. We screened over 16,000 children during their pediatric checkups by enrolling the pediatric providers to partner with us. And you can see that nearly all of them screened negative, meaning low risk for autism, which is what you would expect. When the children did screen positive, we tried to get them to complete those follow-up questions with us on the phone. And you can see that, again, a lot of families who initially reported some risk for their child dropped into low risk when we got more information. Whoops. And then we've tried to evaluate all of the children. Some families did not come for the evaluation. But for the 221 at-risk families who attended, we diagnosed 105 toddlers with autism, and 116 did not have autism. We'll come back to those in just a minute. We also tried several other strategies to find those missed cases, and we found 18 more cases of autism. So if you remember that 2x2 matrix, we can take the bottom of our figure that I just showed you, and in this case, I'm going to stretch it out. There we go. So I'm going to stretch it out to show you the row on the top becomes sensitivity here. The row on the bottom becomes specificity here. And so you can see that with the initial questions alone, sensitivity was very high. Based on the children we were able to find through other concurrent strategies. And it went down a tiny bit with the follow-up, but the follow-up is really important when we get over here to positive predictive value. Only 13.8% of the children who initially screened positive had a confirmed autism diagnosis. So that's not enough confidence that we should be worried about all of the children who screened positive. But when we use those follow-up questions to get more information, that jumps up to almost 50%. So now we can be more confident that a positive screen is really a risk for autism. Specificity and negative predictive value are very high. And then remember the likelihood ratio. Anything that's above 7 is thought to be an effective tool. And so just the first questions alone have a likelihood ratio of 20. And if you use the follow-up, it jumps up to 114. So that means that a child with autism is 114 times more likely to screen positive than a child who does not have autism. So this becomes a very meaningful tool to aid in the detection of autism. So just a few more points about this. So I said that we would come back to those children who screened positive but were not diagnosed with autism. It turns out that the positive predictive value for children with any kind of diagnosed developmental disorder is actually 83%. And if you include children who may not meet criteria for a disorder, but something's not quite typical about their development, it's almost 95% of the children who screen positive on the 2-stage MChat revised with the follow-up questions. So that should increase any provider's confidence that if a child screens positive, they need to go see an expert who can differentiate speech delay, global developmental delay, and autism, so that they can recommend the appropriate services. Looking at the surveillance aspects of this study, we asked the pediatric providers to mark every MChat if they had autism concerns for the child, regardless of what the parent answered on the questionnaire. And the physician concerns only detected a quarter of the autism cases that we found. And in contrast, the screening tool detected 85%. And that suggests, again, that these are very subtle, difficult signs to detect in a checkup. It's not that the pediatric providers are doing a poor job, it's that this is a really difficult task. And then the last thing I wanted to point out about this is that I'm very excited to say that our mean age of diagnosis in the study was 25.6 months. So this is 2 years younger than the national average in the US at the time when we published this. So it tells us that it is possible to detect and diagnose many, many children around their second birthday, rather than waiting until they're 4 years old. So this work led us to come up with an algorithm. So remember I said that a screening tool should tell you how to interpret results and what to do next. So based on the initial 20 questions, if a child's total score is less than 3, then no follow-up is needed at that time unless something else from the surveillance indicates concern for autism. The follow-up questions are most crucial when the child scores in the mid-range. So if there are 3 to 7 at-risk items, getting more information from the parents is crucial. And if they continue to show risk on 2 or more items at that follow-up, then the family should be referred immediately for diagnostic evaluation and early intervention. If their total score is 8 or more, it's okay to bypass those follow-up questions. It would be a lengthy interview at that point, and the likelihood of a child dropping down to low risk is very, very small. So the follow-up questions are really only critical for the children who score in that mid-range. I wish I had time to show you a bunch of these papers, but just to give you an indication, if you go to our website at mchatscreen.com, you can see these references. Each spot on the map represents a paper that I've actually read that conveys something about using the MChat in that region. And so there are many more that I haven't read. There's papers in other languages, and there are some that I think I'm not updated enough to capture on this map, but it gives you a sense of how this work is being done around the world. I seem to be missing a slide. That's okay. So what I wanted to leave you with is some take-home messages. Autism screening is effective at identifying many toddlers with autism. I want to make it really clear that we do not think that every toddler with autism can be detected at 18 months or even 24 months. What I didn't do is show you that our numbers don't correspond to the CDC prevalence data, because we think that measuring autism in children who are 8 years old is different from measuring autism signs in children who are 18 months old. And so one should never assume that if a child made it through a screening, you're done being vigilant about autism. But what we do know is that for many, many children, we can identify their autism symptoms before the second birthday. And if we act quickly on risk scores on screening tools or other surveillance or detection strategies, we can reduce the age of diagnosis by 2 years. And what that does is it gives children 2 more years of the earliest intervention when their brain plasticity may be the greatest, and give them the best opportunities to shift their developmental trajectory. I didn't have time to present this work, but we have some data now showing that some of those disparities that I mentioned in the beginning about children from racial and ethnic minorities or low-income families having later diagnoses than their Caucasian peers, using standardized screening approaches across populations can reduce or nearly eliminate those disparities. And I also want to point out that most of the false positive cases, so children who showed at risk for autism on the MChat, but did not get a diagnosis of autism, most of them have other developmental issues that warrant intervention. And the critical thing to do is to differentiate who needs what type of early intervention. So what's left for us to do? I think there's still a lot of work to be done. So there's an ongoing trial that we're doing right now, trying to directly relate not just screening, but the detection strategies that primary care providers use in their well checkups to positive outcomes for children with autism over time. So we're actually measuring what community providers are doing to detect autism and tracking those children through the diagnostic and early intervention process until they're getting ready to enter kindergarten at age 5. There also are a lot of implementation challenges. So one of the posters that we have at the conference this week is talking about what usual care looks like for many pediatric providers. And only one of the pediatric providers that we interviewed about their detection strategies were doing what we would call best practices, which follows the AAP recommendations in the US of integrating surveillance and screening and acting on positive screens with immediate referrals. Out of about, I forget what our sample was, maybe 50 providers, only one of them was doing best practices. So we have a lot of barriers to overcome to implement these detection strategies in primary care. And then I'm not as familiar with the system here in Canada. I do know that I have a lot of positive expectations for countries that have more socialized medicine, but I know that there are still some challenges. But in the US, our early intervention system is sorely lacking. And there are many, many children who are underserved or completely unserved, even after their autism is detected. And so I think that that can be one of the challenges to recommending universal screening. If we can't treat all the children we'll identify, why should we identify them? And my answer to that is we need to identify them to demonstrate the need. And then we need to fill the need. We need to meet that need by training more providers and giving greater access to those early intervention services. With that, I will stop, and I don't know if there's time for questions.
Thank you, Dr. Robbins, very intriguing. For questions, if I could invite people to come up to the mics.
Hi, you mentioned implementation challenges as one of the things we need to work on in the future. I was wondering if you could briefly go into what the biggest challenges with implementation are right now, specifically if it's more about people with diagnosis, people being screened, getting ineffective forms of treatment, as in not getting the proper types of treatment, or if it's more that of a staffing issue in the actual intervention sites.
So I think that there are numerous challenges. So implementing screening protocols comes with challenges that include providers not having enough training or worrying that they don't have sufficient training to use tools adequately. Time and money is always a factor. I'm not sure if this translates to primary care in Canada, but in the United States, the pediatric provider spends about 8 minutes doing a checkup with a child. And if you think about all of the things that they need to do in those 8 minutes, it's probably not possible. And so time and money is a factor that limits the detection of autism. You also alluded to the implementation challenges at the early intervention level. And so one thing that's interesting to me is how much variety there is in how early intervention is tackled across the different states in the US where I'm most familiar with the system, but also across the world. So there are some places where there are no autism services at all. And then there are other places where the children get sort of a usual mix of speech therapy and maybe a special ed or developmental specialist, maybe some occupational therapy, but they're not getting autism-specific treatment. And then there are other places where I can give an example of Connecticut, because we partner with the University of Connecticut on some of these studies, A child identified with autism gets sent, instead of to the closest intervention office, they get sent to one of 5 or 7 autism centers around the state. And it's a small enough state geographically that they can do this. I don't think that would work as well in Pennsylvania. They would need a lot more than 5 to 7 to cover the whole geographic region of my state. But the idea that these children go to a center where the therapists are trained to work with kids with autism, which is really crucial, and they get 10 to 15 hours of intensive autism-specific treatment is phenomenal. Unfortunately, Connecticut continues to scale back the services they're offering because it's so very expensive. So one of the ways people are trying to tackle these implementation barriers are to investigate alternatives to one-on-one directed teaching therapy approaches. So an example is a colleague of mine at Drexel University is he developed a model to adapt the Early Start Denver model from a one-on-one approach to a small group approach. And he ran a clinical trial in Australia using this approach in a childcare center, where he trained the childcare teachers to deliver Early Start Denver model as part of their daily routine with the children and measure the outcomes. And he's now starting a pilot study validating this approach in Philadelphia. So I think if we can further develop those strategies and also lobby our governments to give more funding to early intervention, we'll start to better meet the needs of the children that we do identify with autism.
Hello. I'm not a professional. I'm a mother of a 5-year-old who has autism, so I apologize if I don't use the right verbology. I'm going to be just a little bit long in explaining. So when I had my second child, who is the child who's diagnosed, our socialized medicine, I'm from here, I had a nurse come and explain to me how to use my breasts to feed my child. We had a nurse come to us and do a whole presentation on not shaking our second baby, which we knew. What we did not have is anybody to explain to us what to look for. From the age of 2, we knew our son was suffering with something. He was not diagnosed till 4. He was misdiagnosed twice. And we could not express what it was because we did not know what we were looking at. So what interventions are being put in place, or is there anything that's being put in place to educate parents, even at the postnatal ward, starting there, or somewhere close to there so that we can know what we're looking at?
I think that's unfortunately not an uncommon path. And, when I mentioned earlier the stress, I think you're really embodying that worry that something's not right and you don't even know who to ask for help. And I agree with you, we've gotten much better at doing early education and disseminating information about certain aspects of bringing home your new baby. And then there are other places where we still fall really short. So one thing that's being done in the US is that the CDC started a campaign, this is more than 10 years ago now, called Learn the Signs, Act Early. And they have a lot of free resources available on their website that can be downloaded by professionals that they then put in their offices for families to see, or even directly by families. And because it's broad about development, you don't have to know to Google autism to find the resources. So just looking up development can bring you to this type of information that can help parents know what to do and where to turn. I think some of the other resources that are out there that are really good are hard for parents to find if they don't know yet that they're worried about autism. So Autism Speaks in the US and also Autism Speaks Canada has some really good resources on their website, but you may not know to go look there. And so I think the primary care providers basically carry the burden of giving parents all the information that they need to raise their children. And so hopefully here in Canada, they give out information sheets about what to expect at this age and that age and that age. I used to walk out with those kinds of checklists at every appointment I took my kiddos to. Mm-hmm. And that way, if your child's not doing the things on the list, you can call the attention to the list and say, you told me to watch for these things by 18 months, but now my child's almost 2 and not doing half of the things on this list. What should I do? And I think it's an unfortunate burden that the parents have to be the advocates rather than the professionals always knowing exactly what to do. But if we can encourage our providers to be more proactive about looking for these warning signs, the parents who are the advocates now will kind of pave the way for the next generation of parents who hopefully will get more information and more educational supports to know what to look for from their pediatric providers.
Just a little fast follow-up. We haven't even learned very much. It's just the smallest bit of knowledge for the parents, and it's now obvious to us, looking at old videos of our son from 2 years old, even before, that he was autistic. So again, it may be disseminating some information throughout, professionals as well as parents would be useful.
Right, absolutely. In my ideal world, I think about the pediatric providers as a bit of a gatekeeper about informing parents about child development, but then all the burden is on this one professional who has a list of 50 or 100 things to do. And so what I would really like to see actually is public health campaigns that not only give pediatric providers tools to inform parents, but what about child care centers? What about the places where you take your kids for like those fun activities like the jumping on the bouncy things or the story time at the library? If there were public health campaigns that put information in all of the places that parents of young children go, it would be more likely that every parent would eventually be touched by these information opportunities rather than restricting it only to the primary care checkup.
Thank you.
Thank you for your question. We'll take one more quick one before we move on.
Yes,
to the next.
hi, Lila Mirali. I'm child psychiatrist, chief of the Department of Psychiatry, Swedish Study here in Montreal. And I would like, first of all, to thank you, and second, to maybe throw a little bit of a ray of hope about changes that are happening presently in Quebec, because there are stories just like the one we just heard about that are making us all sort of feel ashamed for the situation, but there are certain changes that are about to happen, quite significant in Quebec, I believe, and our current sous-ministre of Health, Dr. Lionel Carman, who is a child neurologist, actually went into politics to change the situation with the waiting list. So we're about to deploy in Quebec a universal screening for children, young children, for neurodevelopmental disorders, which is actually a platform that has been imported and validated from the United States. And this will hopefully will give the specificity and the sensitivity has been checked as well to use a combination. The MChat actually is part of the platform, but it is not the only tool used because it is more wide for all neurodevelopmental disorders. So hopefully we'll be able to identify early on some of the signs, the warning signs for early interventions. Now, the biggest problem that I personally foresee is the lack of resources in terms of the early intervention, because it will have to be, evaluated and custom-made, very personalized for each of these children, because of course there are false positives and false negatives at the same time. But I have to admit that this, it also will give us a banque de données, which will be similar to some of the Northern European countries that have very good databases, and hopefully will serve for research as well. So I have to say that this, as well as Dr. Elsa Bach's Navigator project, that's a very, very important initiative looking at offering services, again, personalized accompanying of the families and patients early on to help for the best resources available. I think there are very important initiatives, and we are looking very much forward to any kind of tools that can be developed for early interventions, as well as to hopefully we have been promised an influx of money for early intervention. So again, thank you very much, and I believe that we're probably looking into better days in the next few years.
That's wonderful to hear. Thank you for the update.
Thank you, everyone. Thank you, Dr. Amrali, for that close-up, and thank you, Diana, for a wonderful talk. Help me welcome the next speaker, Dr. Elizabeth Berry-Kravitz.
Okay, I want to thank the organizers for inviting me to talk. It's exciting to be here, and I'm going to see if I can figure out all my equipment. So since I'm old, I come from the days when we used to have carousels, so I'm going to attempt to do this without a computer. I probably haven't done that in quite a few years, but anyway, I am here to talk to you about that piece that Steve Scherer talked to you about when you know all that biology and genetics, trying to move that genetics to the clinic to treat patients. And I'm going to focus on Fragile X syndrome because that's what I work on, but also because Fragile X is something that for a long time we've had a lot of genetic and molecular knowledge to try to do this with. And we haven't successfully got a drug approved for Fragile X because we have seen some barriers and hurdles to this translational effort, and it really is rocket science, just like this little kid with Fragile X here is showing you. And our motto in the Fragile X field is a person who never made a mistake, never tried to do anything new. So we've had some hurdles, and I'm going to talk about that today. And I have a few disclosures, and that's because there are a lot of companies working on Fragile X, and I'm pretty unselective in who I'll consult for. And I think this is a good thing because it means that there's lots of interest and hopefully things will move forward. Okay, so Fragile X syndrome is one of those rare but highly penetrant genes that causes autism, as Steve would say, and it occurs in about 1 in 4,000 males and females worldwide, is the most common known inherited form of intellectual disability, and is the most common known genetic cause, specific genetic cause of autism. We have a simple DNA test that diagnoses it, and it works like this. This is the FMR1 gene. It's got about 10 to 45 of those of a DNA sequence, much like that sequence that Steve showed you up here, but it just goes CGG, CGG, and there's 10 to 45 of those in the normal gene. That sequence is translated into RNA, but is not is transcribed into RNA but not translated into the protein. And so this is the normal situation. You have a protein called FMRP that everyone makes in their brain. In Fragile X syndrome, there's a big expansion of this sequence to over 200 repeats called the full mutation, and the gene becomes methylated. It does not make its RNA. It does not make the protein, or it makes much lower quantities of these, and then that leads to synaptic deficits and intellectual compromise that makes up Fragile X syndrome. So what does Fragile X syndrome look like? There are some physical features that are not uniform but are present in some of the patients: large prominent ears, a long face, large head, sometimes a prominent forehead or hyperflexible joints, and large testes after adolescence. And here's a child who really has most of those facial features, and here's a child who has very few of them. So there's a wide range of the physical phenotype, but uniform is intellectual or learning disability, especially in the males with Fragile X who are more affected because it's on the X chromosome. And the average IQ in adults is about 40 to 50, with a decline with age across childhood, and that's because the Fragile X children are developing, but they're not keeping pace with their typical peers. They have a specific cognitive profile, and the most common presentation in young children is speech delay. They have motor coordination deficits, a lot of behavior problems in multiple areas, and about 50% or so, depending on which study you look at, can be defined as having autism spectrum disorder. Then there's a list of medical problems that we can see in patients with Fragile X syndrome. So the key features our intellectual disability and the behavior problems. And there's a very high frequency of limiting behavior in people with Fragile X, leading to this to be a major economic and family stress impact. And so if we look, we did a study with Don Bailey that looked at how good are the meds that we use in clinic to manage behaviors for supportive behavioral management. And so the green people's medicines are working a lot, and the red people, they're working somewhat, and the blue people, the medicines aren't working at all. So our supportive medication strategies fall short really in every area except for treatment of seizures. And then when we asked the families, we recently did a big survey called Voice of the Patient, and what we saw from families is that the behavior in Fragile X is actually the thing they would like to work on the most in young children, but as the patients get older, the cognitive deficits become more important because of lack of employment and inability to take care of themselves as adults. If we look more specifically at things, the one thing that families would like a drug to target in Fragile X is anxiety. That's the biggest thing. And then second was learning. So we have issues with not having really good behavioral medication strategies and for supportive treatment, and we can't treat the underlying cognitive disorder at all. So we really need to treat the underlying disorder instead of just using the supportive medications that we have now. But to treat the underlying disorder, we have to know what the underlying disorder is. And so we do know, we have the gene in Fragile X. We know that Fragile X results from a lack of FMRP, but what does FMRP do? So FMRP is an RNA binding protein that exists in a complex interacting with all these other proteins. And it exists at the tips of those. This is out in that tree that Steve showed you at the tip of the branch in the dendrite, which is where the dendrite is communicating with the axon on another neuron. And this in red is FMRP. And so it exists out there, and it regulates activation-dependent dendritic translation. In other words, when you get a neurotransmitter activating a brain cell, and then activating the dendrite, and then there is translation of proteins in response, and FMRP regulates that. And we can see this regulation is important because in the Fragile X mouse, this is the normal mouse, and this is the Fragile X mouse, and you can see this mouse has more of these long and spindly, immature dendritic spines here, and it has a higher density of spines, so it's that over-connected type of neuron. And that leads to problems with the electrophysiology, so the communication between brain cells isn't normal, and you get exaggerated long-term depression and reduced long-term potentiation, and these abnormalities actually vary by the region of the brain and the particular neuron that you're talking about. So many years of research, and then we kind of have a mechanism for what's going on with FMRP based on a lot of studies of the type I just showed you. And so I have this nice cartoon that kind of shows you what FMRP does. This is a simplistic version of it, obviously, but anyway, FMRP sits at the ribosome, and it regulates the translation of other RNAs for other proteins. And when something like glutamate binds to an mGluR5 or 1 receptor, there are other receptors that regulate this process too, but this is the most studied one. You get FMRP being kicked off the ribosome and releasing its RNAs so they can be translated into protein. So then you get more proteins being made, and those proteins have effects at the synapse, like bringing in AMPA receptors, for instance. AMPA receptors are the workhorse of the synapse that make you have a stronger or a weaker connection, and so regulating those changes the receptivity of the neuron to communication with other neurons. One of the things that results from this process is that FMRP is made or recruited, and then it acts as the feedback sort of brake on the whole system. So as long as you have FMRP, everything is kind of working here depending on how much input you have coming into this neuron. When you don't have FMRP, you have this kind of constant signaling through here, and you get this increase in these synaptic proteins that are being made, and so you get misregulation of the synapse because of that, and this leads to the intellectual disability, and many of the connections are too weak because of this. And we can show, this is a study at NIH where they showed that there is this increase in protein synthesis that you can see in the knockout mouse, the FMR1 knockout mouse brain, representing that increased protein synthesis because of the excessive mGluR signaling. So whenever you have a mechanism, then you can start to talk about, well, how could we interfere with that mechanism to normalize things? And so when you look at this mechanism that FMRP is involved in, there are a lot of places you could intervene in this pathway. For instance, you could block down these mGlu-R receptors that are signaling excessively. You could interfere with this cascade with the signal transduction from the mGlu-R receptors to the ribosome. You could cut down on the activity of some of these overproduced proteins. You could activate AMPA receptors. You could work on GABA, which will decrease the amount of glutamate in the synapse. You could use microRNAs to decrease some of those excess proteins that are being made. And you could directly block channels that are directly regulated by FMRP. So that's a lot of things, but believe it or not, treatments aimed at all of these targets have actually been used in the Fragile X mouse model. And in fact, all of these have been studied in an early trial, at least one early trial in humans. So we've had a lot of clinical work to try to translate this mechanism. A few of these agents have gone to bigger phase 2/3 trials that could have been used for registration with the FDA. A number of them have failed because of commercial reasons or because of no path forward for development or because the drug had too many side effects or didn't work. And a number in green we're still working on figuring out whether these things could be effective in Fragile X. So we've done a lot of work in the field, and I'm going to concentrate on the two types of agents that went to the big trials and what happened with that. So our baclofen is one of the agents that works on GABA systems. GABA systems are abnormal in Fragile X, and it's related to the glutamate abnormality. And so there's an imbalance where too much glutamate signaling, too little GABA signaling. GABA agonists rescue the death phenotype from eating glutamate in the Fragile X fly. And they also work on the mouse model where they normalize the protein synthesis and normalize these AMPA receptors that are being brought into the cells, and they normalize those dendritic spines that look abnormal and reverse a number of behavioral phenotypes also. So our baclofen is the more potent isoform of baclofen, with racemic baclofen, which is approved and used in spasticity, but our baclofen is not approved, and our baclofen worked better than the racemic form in the Fragile X mouse. So a company called Seaside Therapeutics decided to take this forward into trials, and to go into trials you have to go to the FDA. So when we went to the FDA, they said, oh, you should use this measure called the Aberrant Behavior Checklist, because that's what we're using to get drugs approved for supportive behavioral treatment in autism. So we went with that because there really wasn't any other precedent. And we did a phase 2 trial, and we tried, the primary outcome was irritability, like in the autism trials. It didn't really improve. Irritability is something we measure on this aberrant behavior checklist. But what did improve was the social avoidance scale that came out of a refactoring of this scale specifically for Fragile X that we had done before the study ended. And then this visual analog scale, which was a great scale. The FDA really didn't like it, but it was a great scale where we asked the families, what are the 3 most awful things that your Fragile X child does? Let's rate those during the study. And of course, that came out as significant. But because that doesn't map onto one specific behavior, it was a problem for drug approval. But using this, we went into 2 big phase 3 trials, a trial in older patients, 12 to 50, which did not show efficacy, and a trial in 5 to 11-year-olds, which did show some improvement. First of all, the trial had to be stopped early because the company went out of business. So we were supposed to have 200 patients in this trial, and statistically that's what we needed for our primary outcome, but we only had 159. And then only the highest dose of the drug actually works. But we did have improvement on that dose, significant improvement in irritability and parenting stress, but we had made social avoidance the primary outcome, and the p-value was only 0.08. So this didn't wind up going to the FDA for approval, even though it was really a successful trial. And in fact, if you look at the effect size of that highest dose of our baclofen, it's as high as any of the approved antidepressants that are out there right now for use for depression and anxiety. And then another thing we looked at was the Vineland Adaptive Behavior Scale. We had a small cohort of patients that we had followed for over a year in the open label extension. And the Vineland standard score measures adaptive behavior. The standard score goes down as kids get older because, again, they're not keeping pace in development with their typical peers. This is what the Vineland score should have done during the year these kids were on our baclofen, and the blue is what it did in those 30 kids who were on our baclofen. So we have about 200 patients we could analyze, but the data's at the Simons Foundation, and we haven't successfully figured out how to get it yet. So we need to look more at that, because our baclofen may have long-term effects that we weren't able to study. But there's definitely a responder issue. Some patients responded better than others. This was a kid who literally couldn't get off the bus without having a tantrum, and he had just run a 5K race and gone on Space Mountain at Disney World. So he was a major responder, but there were other kids that didn't respond quite as well. Okay, the other big class of drugs we've had in Fragile X that has been highly successful in the animal model and has gone to big trials is the mGluR5 blockers. So there's this big list of things that they make better in the Fragile X mouse, like the audiogenic seizures that are similar to the seizures that Fragile X kids get as little kids, the epileptiform discharges in the brain, those dendritic spines again, and many phenotypes, many molecular and social and behavioral and cognitive phenotypes in the mouse. In fact, there are over 4 years ago, there were over 40 papers from over 20 labs showing pharmacological reversal about 90% of reported mouse phenotypes associated with the knockout mouse. So there's a very large body of support. There's also 6 phenotypes in the Fragile X fly that were reversed. There's 6 more phenotypes that were reversed by crossing a heterozygous mGluR5 mutant mouse that had half the mGluR5 receptors to the Fragile X mouse. There's a rat now where the phenotypes are reversed. And this kind of stuff coming out in the literature led one of the Fragile X dads in my clinic to come in and say, it's a great time to be a Fragile X mouse. So that's the issue. How do we get this to people? So we did an early phase trial of Phenibom, which is an mGluR blocker, and showed reversal of pre-pulse inhibition, which was something we could measure after one dose. It's a sensory hypersensitivity measure, and it normalized. So we went into phase 2 and phase 3 trials, and what happened in a big phase 3 trial run by Novartis with their drug AFQ-056? Well, this is the placebo arm down here, and lower is better. So the placebo arm actually did better than any of the other arms. But you notice we did a placebo run-in. The placebo arm, when everybody was on placebo, the placebo arm was already doing better. So these were just really suggestible people. In the adolescent study, the placebo arm's in the middle here. But look at that placebo effect. You've got such a huge placebo effect that how are you ever going to see a drug effect? Roche had a similar problem with an anxiety scale that they used in their drug. Remember, these are all behavioral rating scales that parents are rating. So after these big trials, Roche and Novartis both disbanded their programs. But Dr. Hagerman and I in California, we had decided that we didn't like these behavioral scales all that well, and we wanted to measure the Fragile X phenotype. So they let us run in the placebo-controlled trial. Our 2 sites ran eye tracking, where we've shown that Fragile X patients don't look at the eyes very well, and a computer measure of impulsivity, where the patient hits the button when they see something. And we saw improvement in both the eye tracking and this measure of impulsivity during the placebo-controlled trials that failed in the behavioral measure. So what went wrong in these trials? Why did pharmacological and genetic reversal, massive numbers of studies, data from 3 species, target engagement in humans, not translate to successful registration trials in humans? I mean, you couldn't really have any more data than that. So there's some things that might have gone wrong, it turns out. We had, in our baclofen trials, we had big placebo effects, and we used the same inclusion measure to meet criteria for inclusion as the primary outcome. So the families figured this out and gamed their entry by rating the scale higher when they wanted to get in the study. We figured that out toward the end of the study when we found the Facebook site that told you how to rate the ABC to get into the baclofen trial. There's also a subgroup effect. Not everyone responded. The outcomes didn't pick up all the areas of benefit because we were measuring the one behavior. And so the FDA precedent wasn't helping us any, and there were commercial problems with the company. In the mGlu5 blocker studies, those drugs really seem to work on synaptic structure and plasticity, and so maybe we were using the wrong outcome target. Maybe we should have been using learning instead of a behavioral target. We also had huge placebo effects in those studies, as I showed you, and we were using a questionnaire that's pretty variable in the USA in a lot of different countries and a lot of different languages, which is only going to increase your variability. So it's possible the mouse model is not predictive, but I don't think we've completely exhausted our ability to run clinical trials. And so about 4 years ago, this is Fragile X syndrome, we were basically stalled in translation. We had patients, we had a mouse model, we had a nice pathway, we had fixed the mouse, we had run some early phase trials that were positive, and then we had failed in the big trials. So a lot of people said, well, maybe the animal models just don't translate. But I think those of us clinical trialists felt we hadn't done our best job of running the trials and measuring the outcomes, and we were asking the question, can we make it a great time to be a Fragile X kid? And so to do that, we needed to fix the field a little bit. One thing we needed to fix was outcome measures. And so we refactored some of the scales showing that they didn't really work very well for Fragile X because of the intellectual disability associated with Fragile X. We created anchored CGIs, which are clinician global impressions, where the clinician is rating specific behaviors and rating them in a consistent way, and looked at them across different features that are present in Fragile X. We've created a bunch of video-typed language and communication measures where we can objectively rate social and language improvement. We've created we've standardized a number of measures like the KITAP, which is this cognitive computer measure, and the NIH Toolbox cognitive battery, where we can measure cognitive changes on a computer or an iPad. We've even looked at the standard IQ scores. This is the IQ in Fragile X if you use the normal scoring method. We have a Z deviation method where we can actually get Fragile X on a bell curve. And then right now we're developing a behavior app. We've learned that most families with Fragile X come in and rate a behavioral scale on the basis of the last day of what was going on in their kid, but we want to measure over time. We don't want to just know if he had a tantrum yesterday. So we have a behavioral app where they can rate rate behavior in real time on a daily basis that we're working on developing. And all these things will help. Biomarkers would really help, and they're really kind of the rare disease holy grail. We can use them to figure out if the drug is getting into the brain and working. We can use them to figure out whether they correlate with drug levels. We can use them to figure out which agents are actually doing something and move them forward, and for selecting the subjects that are responders. It's best if they translate from the animal models, and we have this thing called ERP, which is an event-related potential. You measure it with an EEG on the head of the person. I'm going to show you that. And then the patient hears sounds, and patients with Fragile X are over-responsive to sounds, and they don't habituate to sounds well, so we can actually measure that on the ERP. And interestingly, we see this increase in the gamma band and decrease in the alpha band, and we can see the same thing in the Fragile X mouse. And this chirp response is a measure of habituation, and we can see that it's decreased in Fragile X and the same thing in the Fragile X mouse. So we can translate directly with these measures rather than trying to figure out whether mouse anxiety equals human anxiety, because probably it doesn't. We have other measures like mismatch negativity that measure the processing of sound, and then we have eye tracking, which measures this tendency of the Fragile X person not to look at the eyes, but to look at other parts of the face. And the ideal thing would be if we could map these on specifically to clinical outcomes, like anxiety and gamma power, or alpha and focus and concentration, or habituation and sensory hypersensitivity, or mismatched negativity and language learning. And so this is a project that we're working on right now to try to map these biomarkers to to different clinical parameters that we can see in the humans with Fragile X. And then so we have some examples of improved outcome measure use in trials. So targeted treatments in Fragile X would obviously modify the whole disease, not just one behavior like the FDA might think. And so what we need to do is be able to measure the whole condition, because there's interpatient variability, and there's interpatient variability in responsiveness, and one behavior may respond better in one patient, may respond, and that may be different with another patient. So we need to measure responses across all domains and positive changes as well as negative changes. So Neuron did a trial about 3 years ago where they attempted to actually measure the whole disease in a trial. And they have a thing called trofinetide, which is a growth hormone analog that works on the signaling pathway between the mGlu-R receptor and FMRP and had reversed mouse phenotypes. So we did a phase 2 placebo-controlled 4-week trial and measured improvement in this new Fragile X rating scale we created, which has all the different behaviors of Fragile X, and a Fragile X visual analog scale where we measured different behavioral domains. And then we did a scorecard for improvement on 5 different measures. And so the rating scale and the scorecard, as well as a number of the other measures, did show a dose response with benefit for trofinetide. Unfortunately, Neuron is working on Rett syndrome right now, so they haven't come back to Fragile X. But this is just an example of how measuring the whole disease was somewhat more successful than using just a nonspecific behavioral checklist. Another thing we've done is with some of this ERP and early cognitive measure development, we can look at some of the early phase trials. So right now we have a trial of BPN 14770, which is a phosphodiesterase 4D inhibitor that raises cyclic AMP levels in cells. And I actually, in my original research in 1988, showed that cyclic AMP levels were low in Fragile X cells. And then subsequently shown in the mouse brain and in embryonic stem cells. And we've been waiting for a drug that would be brain specific to try to raise the cyclic AMP, and now we have that drug, so we're doing a crossover design trial. But we're using these newly developed cognitive measures as well as the biomarker to really decide whether this drug should go forward into a big trial as a go, no-go type of model. And then Ovid has a drug called gaboxidol, and it works on the GABA, the extrasynaptic GABA receptors, and decreases the inhibitory tone again. And so this drug seems to normalize the abnormal alpha in the ERP. So again, here we can use a biomarker in an early trial to determine whether that drug should go forward. We're working on fixing outcome measures, but we probably need to do some work on trial design and developmental disabilities as well. For instance, trial length and the target age. So we can treat the mouse for a very long time in mouse life, but we treated humans who were at least 12 years old for 3 months and thought we would change the whole disease, and that seems a little crazy. We probably need to have longer treatment times. We also need to know some of these disorders you may need to treat for a long time to see change, and so we need to know natural history. So right now we have a project funded where if this is normal and this is Fragile X development, we need to understand in terms of language and cognition and adaptive skills, what is the developmental profile in Fragile X? And so we're collecting that right now so that if we ever had a drug that changed things, we would be able to see that we had a difference. And then another thing is, really, if you're doing your initial studies in adults with a developmental disorder, is it possible to see change? We don't actually know that answer. And in fact, the mouse is an adult. Even if drugs reverse things in the adult mouse, kids haven't even gone to preschool yet when the mouse dies. So we really need to think about these disorders as developmental and do key pivotal trials in very young kids. And the younger kids did do better in a couple of trials. Novartis did a big study and said lack of age correlation for age 12 and up in terms of effectiveness of the drug, but by the time a person gets to 12, the game may be lost. So we need to move younger. And so what we've been doing in the field is this. We've been doing safety studies in adults and safety studies in adolescents, and then trying to start with phase 2 and 3 studies in adults, and then phase 2 and 3 efficacy studies in adolescents, and then trying to get FDA approval, and then going back almost as an afterthought and doing studies in kids. And then later on, even post-marketing, looking at children. So that's only going to work if the drug targets a behavior and targets it relatively quickly. It's not going to work for disease modification studies in a developmental disorder. What we really need to do is these phase 1 studies in adults and maybe use those biomarkers so that we can see that the drug's getting in the brain and changing something abnormal in Fragile X. And then to study disease modification, we need to take a different path. Do some biomarker studies in adolescents. Do the biomarker and PK studies in kids, because these are the things you have to do before you do a big study. then do the big efficacy studies in kids, get approval, do extended studies to see if we can impact the natural history, and then go back and study the adults and find out, can we actually impact a developmental disorder in adults? And that makes more sense. But industry's not really ready for that idea, although some companies are coming around. So another thing we might do if we want to study learning, another thing we might do is embed a learning intervention in the trial so we can see if the drug pushes learning. And there are some learning interventions that have been developed in Fragile X. There's something called PILI, or Parent Implemented Language Intervention, which is a model for delivering speech therapy by Skype, where the parent actually trains to do the therapy with a Skype and Bluetooth device with the speech therapist on Skype. And that was actually shown to improve the use of language in patients who got the PILI versus they had a sham control, basically. And then also CogMed, David Hessel has worked on this and has shown improvements in patients with Fragile X and executive functioning using CogMed. So we proposed about 3 years ago to the NIH to do a very novel trial where we would try to change the paradigm for drug development and neurodevelopmental disabilities. And so we are studying the mGluR5 blocker on language learning in young children with Fragile X syndrome in an effort to really do a plasticity trial and look at the effects of a drug on learning. And this is addressing many of the problems in the prior trials, like incorporating a young age, doing a long trial to study learning. We've embedding a language learning intervention, the PILI intervention, to study and focus the learning in this one area. And then we have objective language and outcome measure and cognition outcome measures with something called the observational measure called the weighted communication scale as the primary outcome, which is targeted to the type of language learning that the kids are doing. And then using biomarkers to try to measure target engagement. And so this is the first ever large multi-site trial studying the effects of a targeted drug treatment on learning. And so the way the trial works is patients come in, they're randomized to drug or placebo. They have a period of time where we find the right dose, and then they start the language intervention, and they do that for 6 months. And then at the end of here, we're going to look at whether the language intervention actually improves their language, whether their language learning is more improved on the drug than in the placebo group. And then they all go on to drug, and we can actually look longitudinally, because the placebo group, we have 6 months with nothing, 6 months with the language intervention, and 6 months with the drug and the language intervention. So we have a longitudinal model as well. These are the sites on the trial. We're enrolling young kids with the Fragile X full mutation who speak English and have intentional communication. And we have we are behind our recruitment expectation here, but we have about 70% of the trial enrolled. We are interested in getting that enrolled quickly, and we are inviting everyone from Canada who speaks English to enroll. So if you know someone with Fragile X, please tell them that we are interested. If they have a young child and they're interested in language, the advantage of this study is that whether the drug works or not, these families get a huge amount of resources in terms of the language intervention. So to do this trial, we've had to create a lot of new processes that will now be available for all neurodevelopmental disabilities for future trials. We've figured out how to do centralized rating for this weighted communication scale. We've figured out a process for getting the SLPs to fidelity in terms of administrating. We've developed standardization of eye tracking and ERP processes. We've created clinical report forms for monitoring behavior and medical history in Fragile X, which was really important because the ones in previous studies had not been well done, and these could then definitely be generalized to other disorders. We've got standardized training of the SLPs for the delivery of phyli and a centralized assessment of all the therapists and parents on the fidelity of delivering the language intervention, as well as we've figured out the use of equipment for Skype and internet-based video conferencing to deliver the intervention and do drug dilution monitoring across all 15 sites. So that's really, this has been a heroic task, and this is going to produce a lot of background work for other future studies. So I just want to change direction a little bit and mention we have a Fragile X Clinical and Research Consortium in the Fragile X field, and this is all the clinics in the USA. We used to have clinics in Canada, but then the CDC funded us, and they said no clinics from Canada. So Now, though, we are developing an international consortium, and I think there's some of the Canadian clinics are involved in that. Our purpose is to standardize care delivery, develop consensus documents so that everyone can have good care, increase research cohort size, collect natural histories, through this project called the Forward Project, and have a clinical trial network. And so one of the things we've done is created a clinical trials committee in the Fragile X field where we try to force industry not to do things that will lead to failed trials because of our past experience in Fragile X. So we're working with anyone interested for free on trial design, outcome measures, the development path. We're working toward more stringent animal work where we want, just if you reverse one phenotype in a mouse, you can't just go into a clinical trial in humans. You got to do this in a couple labs, and you got to reverse an array of phenotypes. And we're trying to mandate a more rigorous early development path. So we do a small study and we demonstrate that the drug is doing something in the brain before we go to a big study where a lot of people participate, and then there's a big sense of failure. And so we're trying to require a discussion with us before a company goes to a big trial in Fragile X. And the National Fragile X Foundation won't endorse them until they talk to us. So I think we've learned a lot in the Fragile X field, and Fragile X is kind of a window to many other single gene neurodevelopmental disorders, because a number of those other high impact genes that Steve talked about are actually in this cascade of proteins that all interact and that interact with FMRP. And so other gene disorders like tuberous sclerosis, neurofibromatosis, PTEN-associated autism and macrocephaly, Phelan-McDermott syndrome with SHANK3, intellectual disability due to AKT, and even Rett syndrome have interactions with this whole cascade. So there's a lot of overlap, and if we figure out how to do things in this pathway for Fragile X, it will clearly lead to work in other disorders. And in fact, the FMRP is kind of an uber protein that regulates a lot of things, and so there's a lot of overlap between the Fragile X pathway and genes involved in autism. There are a number of proteins, some that I just mentioned, that are in the pathway for mGlu-R signaling to protein synthesis that are causes of autism. There's a number of proteins that FMRP regulates. In fact, there's a lot of proteins that FMRP regulates that have been identified in autism exome screens, and it may even regulate as many as a third to a half of those proteins. And then there's interaction. Neuroligin 3, in fact, regulates the mGlu-R receptor activity. So there are these interactions between glutamate and GABA that are related to both Fragile X and autism. So Fragile X is kind of a clinical model in that it overlaps intellectual disability, learning disability, and autism, but also in terms of the molecular events that are regulated by FMRP. And so we've had a number of lessons from Fragile X that can be generalized to other neurodevelopmental disorders. When we started this back in 2002, we were really crossing the bridge. We were really building the bridge while crossing it, and we fell off a little bit, and so now hopefully we can build the rest of it. So FDA precedent's not all it's cracked up to be. It's often unhelpful for mechanism-targeted rare disorders in terms of understanding what the precedents and the outcomes should be. You need to have a clinical trial outcome measure, infrastructure, and sites have to be trained and have good measures and methods that produce consistency. Post-hoc analysis in early trials may be misleading. It's important to develop CNS biomarkers for target engagement and to have ways of running pharmacokinetic and pharmacodynamic studies before going into big trials. You need ways to minimize placebo effects, and so anchored clinical outcomes as well as objective outcomes are really, really important, and you need to manage social media, because if social media goes crazy, placebo effects can go crazy. And finding a way to measure key aspects of disease and not limiting to the one behavior, finding strategies to deal with. If you've got a subgroup of if 40% of people with Fragile X respond to a drug, that's great. I want to have that drug in clinic, right? But it's darn hard for me to show across a whole cohort to the FDA that that drug produced improvement, because I can't run a 2,000 patient trial like they can in depression. So we need strategies for how to deal with subgroups of responders, and this plays into personalized medicine. We need to treat early, we need to treat long, we need to treat young, and we need to figure out how to embed learning interventions so that we can understand whether drugs are really affecting learning in the same way that they do in the animal models. So I think we've been the pilot for doing some of this work, for doing disease-modifying trials in autism and neurodevelopmental disabilities, and the work in Fragile X, I think, has allowed us to learn lessons, to rethink traditional developmental strategies, and to begin to approach natural history and how to prove long-term disease outcomes. And we have had an array of experiences that have already targeted to changes, that have already translated to changes in the way fields like Rett syndrome, Down syndrome, and Angelman syndrome, and tuberous sclerosis are approaching their trials that have followed the Fragile X trials. And so this work has been a catalyst to move into the future with ideas, tools, and hope, and I think that it's a way to approach many of the rare single-gene autisms as they're diagnosed and develop treatments for these. And we collaborate with everybody in the Fragile X field and have funding from a lot of different places, and if somebody's not on here, it's just because I didn't get them on the slide. And then this is our group, our Fragile X clinic group at Rush, and many of the patients we treat. And we are hoping to make their lives better and someday have those FDA-approved treatments. That's it. And I'll take questions.
Lovely. So, questions for Dr. Barry Kravis? To the mics, please.
Okay, I've overwhelmed everybody.
No, that's okay.
And
actually,
they're waiting for lunch.
That's the feeling that I have, is being overwhelmed, actually. I wanted to thank you for your talk. I thought it was really inspiring. I think that it would be great to actually come up with consensus statement in terms of, or guidelines, in terms of looking for biomarkers when they're available, maybe moving away from some of the checklists, I'm a psychologist, I'm guilty, that we typically use that are not really sensitive. And I know that you've been working a lot with SLPs, which is great. In terms of the autism field, we also have, a behavior analysis, which can really help in terms of getting those objective behavioral observations of kids that are doing these trials, and that could be integrated really well. Are you looking at working towards consensus guidelines that could then be, pollinated to, other ASDs, if you'd like?
Yeah, that would be really good for, there was an NIH meeting on biomarkers for neurodevelopmental disabilities, and the idea was that there would be a consensus document that would come out of that meeting, but it hasn't. There was a brief summary paper that was published, but the white paper on consensus has not completely been published. Although there is an NIH statement from the meeting, it's not quite the same as a kind of finalized consensus document. But I think we, as a field, we probably do need to work on that. And I agree with you, there are a number of good social measures that can be videotaped and scored that are really nice. And people are going into the home and trying to count, different kinds of behaviors with home monitoring. And I mean, the field is really, I think is really advancing, and I'm hoping we're gonna be no longer having to rely just on behavior checklists, because we know there's so many problems with those. And they were one way of getting Risperdal and Abilify approved for autism, but now I think we're in a different place and we need to have different things. So yeah, I'll take that back. The biomarker committee that from the NIH meeting, there is a plan to do something like that.
Any other questions? I have a quick question for you on the Fragile X studies. So do you think that the biology we predict at the beginning from the genetic mutation is the same biology that the adolescent and adult mice or humans with Fragile X have?
Okay, that the biology that we predict
based on the genetic mutation is the same. Like, does the disorder change with age enough that the medications we predict from the beginning may not be effective later on?
I think we honestly don't know that. I mean, in the mouse, you can study the mouse as it gets older. And, there are many situations where the adult mouse is given a therapy and it seems to work really well for a lot of phenotypes. And then when you try to do something in humans, you don't get too much. I think the human brain is just much more complicated, and I don't think we know that the developmental windows in the mouse map onto humans at all. We've talked a lot about this in the Fragile X field, and there's a general thinking that maybe we need a primate model. But I think what happens is the FMRP is thought to be a protein that is important both developmentally and on an ongoing basis acutely all the time. So there's a certain amount of things that it's probably doing all throughout life that you could modify later on. But there are also probably some key things it does in development that you need at that time, and then if you pass the window, you might not be able to get that back. So we do think there's benefit to going to the earlier period. In other words, if you wait too long, there might just be some things that just can't be rectified. So what FMRP is doing is probably different at different ages, until a certain point.
Any other questions? All right, so thank you very much,
Okay,
Dr.
thank you.
Clavius.